Profesor Alexandre Soares Rosado. Ahlan wa sahlan, Prof. And the second, Prof. Dr. Patricia King Ji Hung from University Putra Malaysia, Campus Bintulu, Sarawak. And the third, from Brawijaya University, Professor Dr. Agustarno, and the last associate professor, Dr. Suradet Butakon from Kasetsat University, Thailand. This seminar focus on innovative technology in integrated plant pest and disease management. We aim to explore new strategies and tools to improve our understanding and control of plant pest and disease. This event will inspire new ideas and foster collaboration, leading to significant advancement in our field. We also aim to strengthen relationship and build partnership to enhance research and practical application in the future. To conclude, I'm sincerely grateful to all of you, our esteemed guests, for participating in this seminar. I also extend my thanks to our keynote speaker and the organizing committee for their hard work and dedication in making this event possible. Probably that's all from me. And once again, thank you very much. Thank you for all speaker. Thank you for all participants. Thank you for all organizing committee. And enjoy this seminar. Assalamualaikum warahmatullahi wabarakatuh.
Thank you for the head of the departments. Now we are honored to have the Vice Dean of Academic Affairs at the Faculty of Agriculture, Dr. Afifuddin Latief Adirejo, SP MSc, to give the opening remarks on behalf of the Dean. Please join me in welcoming Dr. Afifuddin Latief. Mr. Afif, the time is yours.
Okay, thank you, good MC. Good morning, everybody, distinguished guests, and ladies and gentlemen, professors, lecturers, students, I think, undergraduate students, postgraduate students. First of all, on behalf of the Dean of Faculty of Agriculture, he is now in Germany. We have collaboration, strong collaboration with several universities in Germany. We would like to welcome all of you here. You are here to come to our international hybrid seminar. And secondly, thanks to the Department of Plant, Pest and Disease. We really appreciate for these occasions. This is the first in this year, right? I would like to extend and especially warm welcome to our special guest, Professor, Alexandre Soares Rosado. Yeah, sorry if I'm wrong for you. Yeah, and then Professor Patricia from University Putra Malaysia. Thanks and great to be here. Yeah. This kind of seminar, we would like to advise all of our audience here. Please pay attention every knowledge transfer from the guests, and for the students, please explore everything. Oh, sorry, Prof. Agus, thanks to be here. Our colleague, our professor, as keynote speakers. So my suggestion for you, especially for my student, please explore everything. Please pay attention for all knowledge transfer from them. I think this is good idea to be topic innovative technologies and in integrated plant pest and disease management, because now we are running to become transform faculty. For example, we increase the scientific performance of students. We try to increase the volume of laboratory by constructs and changing the quality and also the quantity. So I think the topic today strongly related with our visions and mission of Faculty of Agriculture. And last but not least, thanks much for organize this kind of seminar, Pak Samsul and team, for Plant, Pest and Disease departments. Thanks, very good. Thanks. You are a very good team, I think. and really appreciate. Hopefully, this is can be continue for the next year, Pak Samsul. Yeah. So because my remark is also to open, we open this, and now officially we can start by Bismillahirrahmanirrahim. Okay. Thank you and good luck for this seminar. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Please give a big round of applause, everyone. Thank you very much for the opening remarks and open this event, the Vice Dean of Academic Affairs. We will now take a brief moment to transitions into our first sessions. While we are in the transitions, and to commemorate this event, I would like to invite all keynote speakers, lecturers, professors, and invited guests to come forward to take pictures together. For all of the lecturers or professors in Plant, Pests, and Disease Department, please come forward. Let's take a picture together. And for all of the committee, perhaps we can take a photos together too. Please come forward. Let's take a picture together, everyone. Especially for all the professors or lecturers from the Plant, Pest, and Disease departments, and for all of the committees, let's take a picture together.
And bachelor degree from Gama Philo University, Brazil, 1989. His research interest is centered around the study of microbial diversity, evolution, and new microbial metabolism using conventional tool associated to multi-omic approach to understand the evolutionary history, ecological roles, and psychological capacity and capability of free, living, and symbiotic microorganisms, including those found at the extreme of where life exists. All right, now let's welcome our first presenter, Professor Alex. Time is yours.
Yes, here. Yes, thank you. Assalamu alaikum. Thank you. Thank you very much. It's a huge pleasure to be here today. It's my first time in person in Indonesia and here in the university. So I thank you very much for this opportunity to showcase some of the work that I've been doing in the last couple of years in Saudi Arabia. I actually presented some of my work last year and the year before online in the cycle of seminars. Yeah, and so it's really nice and really overwhelming to be here in person. You know, to meet you all and to see you all. This is a really good opportunity. I can start my presentation. Yeah. So thank you for this invitation. Thank you for the opportunity to be here, really in person. This? Okay. Okay, so I can try, yeah. Okay, so it's going. No. Yeah, it's going. Okay. It's normal. Always happens with me. So I'm doing a lot of omics and a lot of research, but I don't know how to deal with computer. It's always tough. Yeah, always. My students always make jokes about this. Yeah. So this is a picture of KAUST. KAUST is well, I'm originally from Brazil. I'm Brazilian. I did my PhD in Netherlands. I was taken, and I moved back to Brazil. I went through the whole process to the full professorship in Brazil. I moved to California. I spent 4 years at UC Davis before joining KAUST. and I joined CAUS in Saudi Arabia almost 4 years ago, so 3.5 years ago, and it has been so far a very amazing journey. It's an amazing country. It's a beautiful place. It's amazing people, and it has been a experience of lifetime. So it was a really amazing place. And CAUS is a very international university. I must say this. At CAUS we have 120 different nationalities. So it's a very international research institution. We don't have undergrad students. We only have grad students, so masters and PhD students. We have a lot of postdocs, a lot of research scientists and PIs, the faculty, and it's really dedicated to research. So it's a very, very, wealthy place. It's a very rich university, and they have state-of-the-art facilities. I think I have some of them here in the slides. Yeah, maybe. So yeah, okay. Yeah. So there's the web link, the website on CAUS, if especially the students or anyone interested in know a little bit more about CAUS and about the process to apply for masters and a PhD or for postdoc as well. We have some faculty open positions as well at CAUS in different fields. And as I told you, and I mentioned before, it's a very international place, but we target the priorities of the kingdom that actually are the global priorities as well. So it's a very interesting place. And I think I should point it here. Yeah, so this is just showcase the library. We have the fastest supercomputer in the Middle East and one of the fastest in the world. We have very amazing facilities for we call core labs. So for sequencing, metabolomics, all the omics that you can imagine, imaging like all the microscopes and the cryo-EM and everything. This is a facility for the NMR. So for the metabolomics, it's very powerful as well. So it's very, you have everything there. So there is no excuse to do research and good research. This is why this is something that I always say to my students and visitors. This is literally the view of my my office in a sunset, and we have beautiful sunsets in the Red Sea. So CAUS is very well located in the, my backyard is the Red Sea. It's an amazing place, and yeah, I like to state that. So my background. I'm microbiologist. I like to say that I'm old school microbiologist. So nowadays I'm doing all the omics, doing a lot of genomics, metagenomics, transcriptomics, metabolomics. But what I love the most, what I like the most are the microbes itself. And I do love microbial physiology, biochemistry, and I like to understand the microbes. And my background, especially after my PhD in Wageningen, was soil microbiology. And I have this book. It's not propaganda, not a marketing, but it's a very good book, Modern Soil Microbiology. Just to state that, so my main field has been soil microbiology. But microbes are everywhere and anywhere. And everything is really connected. So it's really difficult to stick in one particular environment, especially when you are a microbiologist. So it's natural to spread, like the microbes. So nowadays I'm actually doing work in totally different fields, always having the microbes and the microbiome in the microbial ecology as the main, yeah, maybe you can help me. Yeah. So those are some of the main research projects and research line that we are developing now in my lab. This is my lab, Microbial Genomics and Biotechnology Lab. So the fundamental aspect, the core research that we do in my lab, it's actually microbial ecology. So understanding microbial ecology and how the microbiomes are assembled, how the microbes interact with each other, and how the microbes interact with the environment and with the host, we can, of course, explore and exploit different ecosystems. And so I'm actually doing work in microbes and microbiome of mangroves using probiotics, so beneficial microbes to increase the plant growth. I will explain briefly in a few slides We do have some human microbiome projects as well, because everything is connected. If you have a healthy environment, we also have a healthy human being, and the microbes actually are inside us. And we have some projects now, and actually this was a special demand from my colleagues from KAUST, because they have a smart health facility over there, and they asked me to help with the microbiome side of this research. So we have some ongoing projects in human microbiome. We have some studies on viromes as well. So the viruses are very important players in the ecosystem. We have One Health, planetary health research, soil microbiome, of course, and some extreme environments, very extreme environments. So the first deep sea hydrothermal vent in the Red Sea that was published actually last year. And we are doing the microbiome research in this hydrothermal vent. Saudi Arabia, actually, it's a quite interesting place. I think everybody knows that it's a quite extreme, they have very extreme environments over there, but it's not only deserts as we usually imagine. If you don't know Saudi Arabia, first thing that came to our mind is the desert. And of course they have a desert, but not one, they have rock desert, sand desert, and mountain desert. So a lot of different kind of deserts, but also volcanoes. They have 2,000 volcanoes in Saudi Arabia. Some of them are still active volcanoes. The last eruption, of course, was like 800 years ago, but we can still see, active volcanoes. We can see the smoke. We can collect and isolate very interesting microbes from those environments. They have hot springs. They have hydrothermal vents in the Red Sea, but hot springs in the terrestrial ecosystem. It's really amazing place to study extremophiles. And as I mentioned, I told you, I love microbes in general. And I have a particular deep passion for, extremophiles, because they are so interesting, so cool microbes. I mean, they can perform in conditions that could be literally, we could die in a few seconds, but they are there, not only surviving, but sometimes thriving, really happy in these very extreme environments. And we have a fundamental research in my lab. So we do a lot of fundamental research, but we also have this applied, arm in my lab. So it's more related to biotechnology, and of course, related to microbial-based solutions. So we do a lot of bioremediation. We have some very successful case studies and application in the real world of bioremediation in mangroves. Before Saudi Arabia, I worked for almost 20 years in Antarctica. That is the opposite, extreme. So it's super cold. Now it's super hot. But they also have snow in Saudi Arabia. That's interesting as well, in the north part of Saudi Arabia. In the winter, we can have a lot of snow actually in the Tabuk region. So it's a very, very interesting place. Yeah. And what we are doing in my lab, we are, as I mentioned, using the whole, possibilities of omics. So using high throughput techniques, so approaches like transcriptomics, proteomics, metabolomics, metatranscriptomics, metagenomics, everything. And we also have we are trying some different approaches to isolate as much as microbes that we can try to isolate. So we are also trying some high throughput isolation approaches that we call culturomics. So it's, it's like the basic microbiology, but we can put this in another level, trying to scale up. So we are trying different approaches as well. And this is good because we can try to isolate different microbes that usually we don't have access to those microbes. As we know, the majority of microbes are not known. I mean, we don't have the microbes isolated. We only know 1 to 10%, of the microbes that we have, and depending on the environment, the microbes that we have. The majority, we just know that they exist because we know the sequences. We can access the sequence of DNA, and so there are some new microbes there. And so we are trying different combination and approaches to isolate or to try to target those, so far not yet culturable microbes. And we have been successful in some cases, but it's really tough, I must say. And what is microbiome? I mean, there are several definitions. There is no consensus. Different groups utilize different, concepts. There is a kind of the people trying to have some kind of consensus, and I like this definition, of microbiome. That is a combination, of the whole microbiota. So we have the compartments. You have the microbiota. So we have the bacteria. You can have the fungi, algae, protists, and archaea. So each one, component is part of the microbiota. But when you add some ecological concepts, that I like this definition this is from Gabriele Berg. It's a well-known microbiome scientist she's from Austria. And this paper is quite interesting from 2020 because actually they put in a room like this one different microbiome scientists from different fields. So human microbiome scientists, insect microbiome science, someone that was working with bats, soil, plants, and they tried to have a consensus. Of course, it was a lot of fight, but at the end, they came with this good definition, actually including the biome concept here, the biome that we have from the ecology, the biome, the whole thing. So you have also the environment included in this definition, and also what they call the theater of activity. So this is the way to try to connect the different compartments of the microbiota, but in the interactions between them, because the bacteria are connected itself. I mean, one bacteria is correlating or exchanging metabolites with another bacteria. So they collaborate, they cooperate in the environment. And also the archaeas, and also the fungi, and also the hosts, so everything is really connected. So when you have the whole picture, it's quite challenging, of course, but I think it's the best definition that we can have so far. So this is a definition that we are adapting my lab as well. And most of microbiome scientists are using this. The microbes, it's okay. The next one, The microbes, they are, especially the bacteria, they are here since the beginning. So 3.8 billion years ago. So they have been evolving in our planet in totally different ecosystems, so they can actually deal with any different place, any different ecosystem, since the volcanoes, hot springs, to soil, to plants, to animals, and everywhere, and including the humans. This is for the students. The microbes, they actually part of our life, and sometimes most of people, we microbiologists, we always think about this, but I know that sometimes, and this is a good thing because even colleagues, scientists, but from other fields, sometimes they don't get the whole picture. So I think it's always important as a microbiologist, try to advocate, improve the microbes and try to leverage the power to show how important they are. So we have the plants, and the plants, they need the microbes to get access to nutrients and the protection and so on and so forth. And other hosts as well. So the whales, we think about the whales, but usually we don't think about those huge whales are eating krill, the shrimp, but they don't have chitinase, the whales. So they rely on a particular microbiota. So they have a particular actually microbiome that can process that kind of food. So the whales are so big and so powerful because they rely on the power of the microbes that live inside them. And the same for camels. That's a good example for Saudi Arabia. So they can rely on grass, they can feed on grass because they have microbes that actually will process the food for them. So this is something that we always should keep in mind. So we are never, and nobody's alone. We always have our microbes helping us. Sometimes they can be a little bit nice and bad, but those are the minority, and usually a disease happens when you have a dysbiosis or imbalance. And there are some other examples like some birds that can eat seeds that contain poison, but they have special microbes that can actually neutralize the toxins. And they're like, I like this example, like the frogs that we have in the rainforest, and some very colorful and poisonous frogs, but usually they have microbes that actually are producing the TTX, the toxin that actually are poisonous. That is the same toxin that we find in the octopus, the blue ring octopus. That's the only poisonous octopus that we have. And actually, this octopus is poisonous because it contains special, particular microbes, bacteria, that are producing the toxins. So life is always evolving together with microbes. That's my point on here. And this is also interesting because trying to be not the center of the universe, but I mean, we humans, we also rely heavily on the microbes that live inside us. They are so important, and now the microbiome field is showing this every day. We have new publications showing the power of the microbes in several different aspects in our health, in our also disease. So it's not only like the Mycobacterium, tuberculosis, or Staphylococcus aureus, so those microbes that we know that can cause disease in some particular situations, but everything, even our mood. If you are feeling depressed, there are actually connection with the microbes in your gut. If you diabetes, obesity, you know, some of especially the modern society diseases that we have are really connected with the microbes that live inside us. And there is a connection with the diet, there's a connection with the health of the plants, the connection with the health of the ecosystem. So everything is really connected. This is really important. And I'm showing this slide because I like this definition of Apana from 2008, that actually it's very extreme, but it's quite interesting. He called it like an ancient human. So it's pre-2007. So this is before the next generation sequencing machines. And so before the knowledge that we have nowadays about the microbiome. And what he calls the human, the modern human. So now we know more about the microbes that live inside us and in our skin, in our gut, in everywhere. And we know that they have a special and really important role in our everything, in our everything. That's the truth. Okay, it's easier because, yeah, next, please. Be faster. And the microbiome concept, and especially when you think about One Health, it's so important nowadays that even totally different disciplines like architecture, I mean, some of them now, some schools of architecture, they start to incorporate or try to incorporate the concept of health microbiome to health cities. So you can have health buildings and health cities in the end. So it's a whole new concept, and that's beautiful to see, you know, the microbes really spreading, you know, the wings, not only to the hosts or the ecosystem, the environment, but also the way that we will think about our cities in the future. So this is very interesting. There are some projects. I have some colleagues, microbiologists in Australia, helping companies, architecture companies, to incorporate the concept of health microbiome in the architecture, so you can have better parks or buildings, the way that you construct the building. So, you know, to have a healthy and happy city. This is so interesting. Next, please. And everything comes to this One Health concept. That is actually not new. Aristotle, the Greek philosopher, was the first one to use this overall idea of One Health. But since the last decade, it's becoming very important because the United Nations also adapted this concept. So, and this is good because so especially the policymakers, they can pay attention to this overall picture. And we have a lot of layers, different layers of health. So you have this everything started with the public health. We have the global health and the One Health. So the ecosystem health, and now we have the One Health, connecting everything, the human health and the ecosystem health. So the plants, the animals, and the ecosystems as well. So this is One Health. And we have a new layer. That is the new kid on the block. Is the planetary health. It's from a few years ago, 5 years ago, something like that. Actually, everything is very similar. Planetary health also tried to incorporate some climate change, concepts into the framework. But the thing is, the One Health of animals, humans, and our planet is all microbiologically connected. So the microbes can have a capillarity in everything. And this One Health has grown this scientific conscious over the past decade, as I mentioned. And this is good because now we have several centers dedicated to One Health. And this is good because we can connect with different expertise as well, and not only think about one, little box. We can spread even cows now. In cows, we have one of the centers of excellence that we have now, starting now on July, is dedicated to One Health. Smart Health or One Health concept as well. So we have experts from the microbiology, medical doctors, some people developing some devices as well. So it's very interesting, broad concept. Please, next. Next, yeah. And we have a lot of challenges. And I ask someone to control my time, because when I start talking about microbes, I can lose a track. I always forget to. I really love those tiny creatures, yeah. And we have a lot of issues that we need to deal with, for the next decades. We need to feed a lot of people. We have a growing population. We, of course, spoiling and destroying our ecosystems, and we have a lot of issues that we need to reorganize our way of thinking, and that's a big issue. I think the microbes, they can help us, or they can, to try to deal with some of the biggest issues that we will face and we are facing now. So we can actually leverage the power of microbes to help us to fight climate change, to fight poverty. We can use more, the power of microbes to help us to feed this huge and growing population that we have. Of course, not only the microbes, but the microbes can play a very important role in the equation. And we must include the microbes into the equation. So this is something that we've been advocating for a while, but it's becoming stronger and stronger now. And for the first time, and this is also a good thing for the students as well, it's always good to be part of a society, a microbiology society, any society, because so we have more voice. And now microbiologists are getting more places to speak about microbes and the power of microbes in conferences like the United Nations conference, COP, conference on parts, climate change, biodiversity, so because we must include the microbes. We cannot talk about biodiversity without the microbes. We cannot talk about climate change, not paying attention to the microbes, because the microbes are the main players in most of the biogeochemical cycles. So how can we talk about, carbon sequestration without microbes? That doesn't make sense. But that's just good, because so there is a huge movement now. I'm part of some societies like ISME Society, International Society of Microbiology, MMI, Applied Microbiology, I don't know the names, yeah, it's International Society. And we are really pushing the boundaries, trying to leverage the power. And I'm glad to say that we just founded now the first Middle East Society of Microbiology and Biotechnology. I was one of the founders. And this will be very important to really spread the word of microbiology in the Middle East as well. Next, please. And knowing more and better about the microbes in the microbiome, we can have, and including the One Health concept, we can have more precision treatments. Like for humans, we know about the fecal transplantation, probiotics, precision medicine. So everything is really connected. When we start to know more about microbial-based solutions, especially the probiotics are very important in this one, but some other microbiome-based solutions. And the same for soil, for agriculture, like soil management. We can use single inoculants that we've been using for a while in agriculture, but now we also can explore more the combination of different microbes as well. And this is really evolving. It's really, I have a lot of gray hair, white hair now, but it's really, I feel really young when I start to discuss about this with my students. So I feel, wow, that's so good to be alive at this moment now, especially with the tools that we have nowadays. We can really, wow, this is the feeling, wow, every day. It's really overwhelming. Please, next. Yeah. Yeah, and the microbes, the microbiome, they can help us with several different things in bioremediation, in food processing, production of biofuel, and so on and so forth. And this is just to, we published this paper in Nature Microbiology in 2022. It is a perspective paper, and this is also mainly for the students in the audience. If you want to know a little bit more about how can we explore and harness the microbiome power to prevent global biodiversity loss, this is a very good paper because we have different experts as well, including Gabriele Berg, the one that defined the concept of microbiome, and some other colleagues. And we put this together, the idea of a health environment. And when I talk about environment, I include plants and soil and humans, any ecosystem, and what is a ecosystem with disease, and how we have this, thing. So it's a very interesting paper, but I will not go in details because I can spend a lot of time this. Next, please. Yeah. And nowadays there are a lot of ways to try to build synthetic consortia, SynCon, synthetic microbial consortia, so to engineering microbiomes. We have some processes that are bottom up, so we can really use genetic tools or CRISPR-Cas to modify some microbes and try to study these in the ecosystem and see how this will go. And we also have a lot of top-down approaches like normal enrichment culture or artificial selection, direct evolution. So there are several processes. And this, what I like in this slide is that most of those processes are actually, especially the top-down approach, are based on basic microbial ecology rules. So the microbial ecology is really coming, to this direction to help the development of, the microbiome modulation field. So it is really interesting. Next, please. And this all came with the natural microbiome. We can use the omics to know better about those microbes. We can use a lot of bioinformatics tools nowadays to study the network interface. We can predict functions of those microbes. We can try to understand, to modulate, to model the interactions between different microbes. And we can go to isolation using those top-down approaches that I just touched base in the previous slide to develop a good or potential good consortium. And we can apply this in first in the mesocosmos or in greenhouse. And if works, we can go to the field. So this is the overall approach that we have nowadays. But the thing is that we have tools now that we can really process this framework in a nice way. We have bioinformatic tools that we didn't have last year. So this is always changing so fast. We have, of course, a lot of omics tools, including the culturomics as well. And we have ways to really follow the path of those microbes or the consortia in the field. And we have so fine-tuned tools that we can really try to follow the path of one strain into a complex community. Not there yet, but I mean, we are getting there. Next, please. So it's really, really interesting. And after decades of metabolomics, genomics, and omics, we really have a different momentum now, to discuss about this. And these microbiome solutions are really take over now. They are really getting very important, because a few years ago, it was basically something for the academics. So we had a lot of discussions about the pros and cons, the potential. But nowadays, it's becoming a reality. I mean, we have really powerful tools to really leverage the power of those microbes. And I'm talking about a huge amount of money in investments. And usually when you think about this, money is what they like to hear, the policymakers and, the things. And that's something that we can use in our favor. I mean, also, we can show, wow, the microbes can really help with this. Yeah. And next, please. And just a brief overview, this search for some of the top startups in Europe, in US, a few years ago. And the top 5 actually are microbiome-related startup companies. So this is saying something that's really, overtaking, the field. It's really getting a good momentum now. So it's not only the fundamental research that is exploding, that is really interesting, but also the application and also the money-making stuff of this, that is really good momentum. Please, next. I did a lot of previous work in manipulation of microbes and microbiome modulation. I mean, it's better to say microbiome modulation, because when you say microbiome manipulation, some people don't get the idea, say, well, manipulation doesn't sound very good. So it's modulation, This was a case study from several years ago in a mangrove. In that case, it was a heavily polluted mangrove. There was a huge oil spill, and they tried to revegetate it, to reforestate the mangrove. But of course, the mortality was like 95%. They couldn't replant, they couldn't regrow any plant over there. And so we came with this bioremediation solution. So we combine actually microbes that could eat the oil, different fractions of oil, plus microbes that could help the plant growth, with plant growth promoting features. And the combination, we test several different combinations, several different things in the lab first, in the greenhouse after, and finally we got the chance to go to the field. And actually, next, I think the picture shows better than anything. So this was before and after our bioremediation approach. Before, almost nothing, everything was dying. And I have a colleague from Australia, Norman Duck, and he published a analysis of publication papers showing that if you have an oil spill in a mangrove, and if you leave the oil there, the mangrove will take 25 to 30 years to recover itself. With our approach, using microbes, we could we start revegetation in 2.5, 3 years, so 10% of the time. So this is really making the difference. Yeah, next, please. Don't know my time, but please, when. So Saudi Arabia. So when I moved to Saudi Arabia, as I mentioned before, it was an amazing surprise, because here we can see what they call the harats. Harats are the volcanoes that we have in Saudi Arabia. So they have a lot of beautiful places with very amazing volcanoes. And the huge deserts, sandy deserts, and also hot springs, and the Red Sea, that is also impressive, amazing place. And next, please. Yeah, some pictures that we can see. We can see the Red Sea and some of the deserts that we have over there. The first one is going to north, and the red one desert is actually my favorite desert over there, because it looks like Mars, actually. is in the north region of Saudi Arabia, in Tabuk region. It's beautiful, beautiful. You feel like amazing there. Next, please. And here we can see some of the volcanoes, the white and black volcanoes, the hot springs, the volcanoes. Yeah, sandy desert in the empty quarter. There is a huge, like, like 1/4 of the Saudi Arabia. It's a huge sandy desert with huge dunes. Also, very interesting place to isolate microbes. Next, please. And when I think about extreme environments, actually, we don't know the limits of life. We don't know yet the limits of life. We have microbes that can thrive at 122 degrees Celsius. So if you put in the autoclave, that bacteria or that archaea will be very happy. So autoclave, we have 121 degrees Celsius plus the pressure. But we have microbes that they don't care about this. They're just, whoa, this is home. Yeah, so we still don't know the limits. The same for low temperature. I did bioremediation applied work in the Brazilian station after the fire that they had. The station was burned to the ground, and there was a huge diesel spill. I was doing fundamental research, studying the microbes in Antarctica for a while. So the government, they asked me to put in place a protocol for bioremediation because they knew about my work in mangroves. I mean, different, but I mean, okay, let's try this. And after several trials, we came up with a good consortium of microbes that could perform bioremediation. So they could degrade the diesel fractions even at -20 degrees Celsius. So some microbes can actually metabolize, be active in -20, sometimes -25, depending on the microbe. So it's really, we don't know the limits of life. We all know the best example of the power of the thermophilic microbes or the extreme microbes are the thermos aquaticus, the tachypolymerase that we have PCR nowadays. That's, you know, everywhere in medical field, in microbiology, in agriculture, in any place. But actually, the microbes from extreme environments, they are actually helping I like this example because we have the Thermus aquaticus, the bacteria, that was described by Thomas Brock in 1969. After that, Gary Mills, developed the PCR reaction, get the Nobel Prize for this. And in the same pound, the same hydrothermal hot spring in the Yellowstone, in few years ago, some scientists from MIT, they isolated one fungi. And this fungi actually, it's producing a lot of biomass. And now they are using this to produce food. This is fine. It's a startup company, but actually it's a startup that now they raise a lot of money with this idea using fungi, microbial-based protein. That's interesting because you can use less area. We don't need a huge area. They have a factory or something like that, and it's quite interesting concept. And it's becoming very popular in some places in the US. They find you can buy in Holy Grow food and something like that. So it's quite interesting. And they raise a lot of, talking about money, they raised like $500 million now. So it's not a small startup anymore, because the idea to use microbes, protein, and food actually catch the attention of NASA, the space industry that is also growing now. And this is a potential good solution. And I don't know if you are following this, but now there are huge race and discussions about establish a colony on Moon first, but on Mars in the future, and including Saudi Arabia and the Emirates also, they are joining this idea. And so this is a potential way to have food for the colonizers in the other, moon or space. And so they raise a lot of money, this startup now. So it's becoming a really big business. Next, please. Okay, so I really real rush here. Yeah, so and in this time we built a huge collection of microbes, and now in Saudi Arabia as well. So we have more than 2,000 isolates, and we know the isolates, we know the genomes, we know the everything. So So those are isolated from very different ecosystems. So we have novel enzymes, novel species. Next, please. One example is this microbacterium. So we have a new sunscreen because the bacteria is there producing carotenoids, and we isolated a new carotenoid. We tested this in human cells, and we team up with some colleagues from the chemistry industry, and they tested different oils, and we have a patent and we have a product. So this came from the extreme environment, and actually very efficient sunscreen, 50 times better than the commercial sunscreen, and natural actually. So it has less impact for the environment. Next, please. We are doing the same in Saudi Arabia. We are isolating a lot of UV resistant microbes, so they are resistant to radiation. And this is quite interesting for astrobiological and space companies as well. Next, please. I'm trying to go fast. This is my postdoc, Junior. She's actually leading these stemophiles and novel carotenoids and this kind of work in my lab. We have a lot of novel species, novel carotenoids, novel pigments. So it's a huge potential. Next. I try to give an overview of some of the work that we have in the lab. And this is the consortium that I was talking about today. So a very interesting consortium of microbes, very simple, but very unique. It was a consortium that was growing only using gas, trace gas. So we don't add any organic compound to those microbes. No glucose, nothing. Only carbon monoxide, carbon dioxide, can be hydrogen and nitrogen. Only gas. That's it. In this consortium, in salt and water, nothing. No other source of food. And there was a consortium, very oligotrophic consortium growing this. And say, well, let's see how this will go. And after 3 years, the consortium was very simplified, so only 4 microbes. So those are the metagenomes, the mags of those, the consortium. And actually, one of the microbes, one of the bacteria is one high-GC bacteria, is actinobacteria. And we published a paper last year showing that actually a new family of bacteria. So it's not even a new species, a new genus, it's a new family, Carbonoactinosporaceae. And the next thing, we published a paper there. That's me without beard. Sometimes I like to play in the lab. I really am a lab rat guy. I love the field and I love the lab as well. I don't like very much the meetings, but sometimes I know that it's obligation. So, but I try to be in the lab as much as I can. It's difficult, but I try, and I did the isolation myself. So I'm very proud of this slide because, so the main player in that consortium was difficult, but I was able to isolate based on the metagenomic data, some combinations of, culturing conditions, and I isolated the main player, the actinomycete in the consortium, and we are finishing this data now to publish, to submit the paper, and actually it's very unique. The bacteria contains 3 carbon fixation pathways. In nature, we know only microbes with 1 carbon fixation pathway. So the way that some microbes can catch the carbon dioxide and transform this in organic compound. There are some reports on literature on the hydrothermal vents that based on metagenomic data, not on isolation, that some microbes maybe could have 2 carbon fixation pathways. But now for the first time ever, so far, we have 1 isolate. And actually we're showing that this microbe can fix carbon using 3 different pathways. So it's a very versatile microbe. And this can change a lot of the way that we think about carbon fixation in nature as well. We have proteomics, we have transcriptomics, we have the genomics. I have a nanosim, so this lighting microbe, hypha here that you can see in the below in the slide, is the incorporation of carbon-13, the isotopic version of carbon. So we proved for several different ways that the microbe is actually active in fixing carbon in different pathways. So it's quite amazing paper work. I like that. Next, please. Yeah. Yeah, it contains carboxysomes as well, and that's also new stuff. This is also one paper is under review, and the other one is related to the carbon fixation pathways. I plan to submit when I come back. Please. Yeah, next. We are doing work in mangroves in Saudi Arabia. So we built this mangrove microbiome initiative. That's also good. We don't have partners from Indonesia, not yet. We have some colleagues from Malaysia that join our group. It's a global initiative. So we have people from everywhere with different expertise, mainly related to microbiome. Next. I'm almost there. Yeah, so those are some of my postdocs working in the mangroves, please. Again, some postdocs. So we are also in Saudi Arabia now. There are some huge initiatives to grow, to reforestate mangroves in the Red Sea coast and also in the Gulf Coast. And most of the places it's going well, but in some places they have issues with some fungi that are causing diseases in the nursing of the mangroves. In some places are actually quite tough for the mangrove trees to establish. So we are helping with the microbial solution. So we are leveraging the power of probiotics also to help in some of those projects in NEON and also Aramco now. And so those are some of my students and postdocs. Hamad and Hani actually are the Saudis leading this overall mangrove study. Next, please. Yeah, we also engineering using the same top-down approach that I mentioned before. In mangroves, we are now tackling the microplastic issue and using the same approach, ecological approach, to try to build some synthetic consortia, something like that. So I have Diego, is my research scientist leading this microplastic stuff, research in my lab. Diego is from Colombia. He joined my lab last year. And the main student is Leila. She's a Saudi student, and she's a very powerful woman. She presented her work last year in the L'Oreal Prize for the Middle East, and she won the prize, the first place. And she's super happy, and she's doing amazing work in studying this consortium of microplastic degradation. And seems that we have some novel enzymes as well. I'm almost getting there. Next, please. Yeah, we are doing some work on biocrust as well, imported for Saudi Arabia. Next, please. Yeah, I have a lot of slides, but yeah, but I go fast. Yeah, next, please. Yeah, we are studying also. We are applying some penibacilli to try to improve plant growth. Next. Actually, my PhD thesis in Wageningen was with penibacillus to increase plant growth. And we are exploring different ecosystems, very different, like the Amazon rainforest. I have a collaboration in Brazil, and also Sahara deserts, different plant species and different bacteria species. Next. And one very interesting thing that we have now is the Dark Earth project. It's in Amazon forest, in the rainforest in Brazil. It's a very actually poor soil. And but the forest is very overwhelming. It's amazing. But the forest needs, the forest to keep going. If you remove the forest, if you put down the forest, the soil will be exhausted in a few years. And so it's not a good idea to put down the forest, what they were doing before. And but despite this poor soil that we have in the whole Amazon region, the whole rainforest region, there are some spots, hot spots of very, very fertile soils that they call dark earth. Next, please. And I have a student now, Anderson. He's a Brazilian student. He's in my lab now, studying these dark earth spots, and it's actually pre-Columbian soil. I mean, it seems that the first human civilization people in the rainforest, they start to build and to construct, to help to build these very fertile soil. But the thing is that we don't know much about this kind of soil, and we don't even know even less about the microbes in the soil. So this is what we are doing now in the lab. Next, please. Yeah, it's a very huge, rich soil in organic matter. It's really dark. Next. Yeah, please, next. Yeah, sorry. And we are trying to do kind of microbiome transplantation, you know. Next, go faster now. Yeah, and we have some very good results showing that actually when you use the soil, the dark earth as a simulant on a poor soil, actually can increase the plant growth. Next, sometimes 7 times, further better. And this, of course, is because the nutrients in the dark earth, but also the microbes. And we have some data showing that they have a particular combination of microbes that we had no idea before. Next. This is what we are studying now. So this is Anderson in my lab using also the omics, but he is one of the culturomics approach. He's using this eye chip. So he can put this in the soil and leave there for weeks. And after that, you can recover a fraction of microbes that usually we don't recover doing basic microbial, isolation techniques. So he's really spreading the next, please. Yeah, and this is getting, of course, the news in English, in Portuguese, and even in Arabic. And so it's really getting, because it's so interesting. I mean, you have the rainforest helping the fertility of arid soils, like in Saudi Arabia. And next. Yeah, and this is Isabella, is a student, and she's testing Penibacillus. So I went back to the Penibacillus and Bacillus. And here's a combination of new strains of Bacillus thuringiensis and Penibacillus. Bacillus thuringiensis, this strain is very powerful for biocontrol. We analyzed the genome and all the data. And it was a very good combination with this Penibacillus polymixa strain Cap238 that actually hold very good plant growth promoting features. And the combination actually leveraged the growth of this Tropoellum majus. There is a flower that you can eat. There is in Brazil, and actually it's an unexplored environment ecosystem. So we isolated these strains, this bacteria from that plant, and actually quite interesting. And next, please. And now we are testing. Yeah, next. Otherwise, yeah, next, please. Yeah. Yeah, so it's a different host. And next, I think the next one I can show. Yeah, so those are the characteristics of those strains. And the Penibacillus brasiliensis as well. Very important. It is also new because, I mean, there are not many studies on this group of microbes showing the potential for biocontrol as well. Next. And I want to show, yeah. And now we are testing those strains, novel strains, if they can help the growth of tomatoes using soil from Saudi Arabia, so arid soil. And this is still ongoing work. And so far, seems that actually they can help, at least in some characteristics, to help the plants to keep and to cope with the arid conditions, drought, and also the salinity that we have in soils in Saudi Arabia as well. So this is a very interesting project that we have now in the lab. And next, I think sorry. Yeah, Mars in Saudi Arabia. And what is the next, please? And the connection, yeah, a few years ago, we published a paper, a provocative paper, show, wow, if you want to discuss space, if you want to conquer the space, the NASA, the Chinese space agents, the India, and European space agents, they must include the microbes in the equation. I mean, we of course need the rockets and the satellites and everything, but if you want to really build a colony, if you're really serious about this, you will need microbes. And we published this paper, actually become very popular paper about inevitable future, space colonization beyond Earth with microbes first. And actually, it was provocative paper, but now some agents are Next, please. Asking about this. And so we started a collaboration with South Space Agents and NASA, and we are using now Mars regolith. So we don't have soil. There is no soil in Mars. It's a regolith, no organic matter at all. It's very, very poor, very difficult. If it is difficult to grow crops in Saudi Arabia, on Mars would be it's impossible. And so our idea, well, if you have some interest in microbes, let's study if they can improve the regolith condition. And with some other approaches, maybe in the future, you can use this as a substrate to grow plants and for the astronauts to colonize and stuff like that. And of course, this is one way in space. But what I really like is the way back, you know, when you raise the bar and you have real difficult challenge, I think it'd be easier to apply this in Saudi Arabia, you know, that not easier, but I mean, easier than Mars. Next, please. And we are studying some of the microbes. This is one fungi that was isolated from a volcano in Saudi Arabia. It's a black fungi, a lot of melanin, but this is, it's extreme fungi. We have 2 papers submitted, it's under evaluation now. And we are studying the metabolomics. So what these fungi is producing, the metabolites, when we add this in Mars regolith. And actually, this one is quite interesting because can survive to huge doses of radiation and huge concentration of perchlorate. We don't have many perchlorate here. We have in some volcanoes. And perchlorate is incompatible with life. I mean, we and the plants, we cannot cope with perchlorate, but this fungi can help us with this because it can tolerate a huge concentration of perchlorate. So it's quite unique. This catch the attention of NASA because so they have a potential good astrobiological model to study. It's better than the fungi that they are testing now. So it's really unique one. So we are studying this. This is Aleph, my student. Next, please. Yeah, we also have some new novel bacteria, Halobacterium from Al-Haba crater is a volcanic crater in Saudi Arabia. Also very extreme bacteria that can tolerate a huge concentration of salt as well. We are studying also the metabolomics of those microbes. So the idea here is try to, improve the soil condition using those microbes first. So checking if they can produce some metabolites, if they can improve the aggregation of those regoliths, so if they can add some organic matter, so they will pave the way, that's our idea, for future application of this kind of substrate. Next, please. And to finalize this, so we decide to combine everything. So we have now also the dark soil from the rainforest with Mars regolith. And surprisingly, actually, there are a lot of novel microbes, not surprisingly, but I mean in the dark earth soil from the Amazon forest, the rainforest. And I have this student, it's a visiting student from UK. His actually background is astronomy. And he said, oh, I would like to work with astrobiology. And there is a huge demand now in Saudi Arabia also for the South Space program. And okay, so let's play with this, but let's try to apply this in not only the astrobiological thing, but let's try to apply this in some maybe useful thing for the astronauts or for something like that. And actually, the preliminary data are very interesting. I mean, it seems that the microbes in the dark earth is improving the condition of the Mars regolith as well. And let's see what's next. Next, anyway. Okay, so finish. Yeah, next. We have been in my simulator. I think I'm getting there. Yeah, next. Just showcase, we have a paper that was submitted today describing 26 novel species from, so this collaboration start and become stronger with NASA. So we are helping to isolate novel microbes from the facilities and the Phoenix mission and something like that. It is quite unique. I mean, it's a very extreme environment actually, because they have a clean room. And we have 26 novel species in one sample, a small sample. So it's quite amazing. Next, please. And I think it's the last one. Yes. Yeah, so this is part of my lab. I couldn't have everybody, so I was without beard there. My kids are complaining about my beard. My mother as well. She thought that I'm looking older than my mother. But yeah, And this is my Twitter account. The lab social media is important nowadays, especially for the students, and Chaos likes very much the social media. So, and also our website. And thank you very much, and sorry for my time. Yeah, thank you.
Okay, thank you for that wonderful presentation about microbe and how important microbes in our life. Okay, now let's please welcome our next presenter.
Thank you so much, Ms. Chairperson. Thank you for the organizing committee for inviting me here. It is really a pleasure. I really like Malam very much. Thank you for everyone that joining us today. I think this is a good arrangement. We have Prof. Alex to talk about microbiology. I'm also a microbiologist, and to hear talk about the amazing thing about microbes. I remember we are actually quite experienced, or if I wouldn't say old people, but it's when we started with microbiology, when people ask us, what is microbiology? And then we try to explain things. For them, they will lose interest because they cannot see. It is, you can't see with the naked eyes, right? But when people talk about, oh, actually, I'm a biologist, I'm looking at the birds, everybody get excited because you can see animals, birds, plants. But when you talk about microbes, what is microbes? Nobody interested. But nowadays, because with many tools, new tools, we not only just the microscope with the sequencer, so people can see, well, there are some spiritual things there. We can't see them, they make a lot of impact to us. And I'm sure nowadays everybody can feel the strength of the microbes after the COVID, kan? Selepas COVID, everybody knows what is virus. Before this, maybe nobody understands what is virus. So today my topic is about old wisdoms and new tools. Whatever we know is actually old wisdoms. But how, with the new tools, can we do new things? So redefining the integrated pest management. I think today's topics that selected by the organizing committee is very important, integrated pest management. Prof. Alex had talked about the amazing things that microbes can do, and he started to move away from planets to the Mars. So you know why? You all know that these days it was very, very hot, right? It has been the 12 months, which is really, really severely hot, and the temperature, average temperatures of the universe, our Earth actually increased. When I look at the presentations of Prof. Alex, I just feel like I want to go for holidays. There's so many great places, desert, things like that. But I can tell you, not too long, if we don't do anything, we will have desert here in Malaysia and Indonesia and everywhere, because the climate change is so intense, right? And myself, I'm a regenerative farmers practitioner. What I believe is that we only have one Earth. Before we can move to Mars, before your study actually have some result, we only have one Earth at the moment that we can live. So we can't plant Mars there. But we hopefully one day our next generations will be able to survive in Mars. But now we only have one. We only have one planet here. What can we do? So I really like the what has been shared by Prof. Alex. There are many microbes that can do wondrous things. They can do in Mars, maybe in Saudi Arabia. They definitely can help us here. Why this is important is I would say that you all can really feel this. The agricultural land that is very fertile is getting lesser and lesser. And there's not much things we can do about it unless we make it healthier. So there is no, remember that last time we have farming, and then we have this organic farming. Organic farming basically is to maintain the health of the soil. But we cannot just maintain anymore because they are sick now. They are not healthy. They have a lot of heavy metals. That's why the bioremediation come in. So can we do how to make this better? And with the with the climate change, we also have experienced more pest problems, right? I'm sure that a lot of farmers will complain that they have more disease because of the floods, more fungus disease, and then we suddenly have a lot of pests just wipe out our crops, and the nematodes, the root nodes, nematodes problems, all this. Can we do something about it? That's why this topic's about. I have to control the slide. Is there a control? I just said next. Okay, I will just say next then. Next then. Okay. Just want to say thank you for joining us today. Thank you. Next. Okay. Just want to say that I'm a believer of that a sustainable agriculture practice so that we can meet our current food and textile needs. But however, whatever practice that we have, we should not compromise the next generation's abilities to meet their needs. That's why it's sustainable about. We cannot be that next. We cannot be that selfish next. It's because I don't want to look at my face on next. So basically, we are here. People believe that we are here so that we can pass down our genetics, so that we have next humans and so on. Therefore, if we just thinking about this generations, then that's not enough. We have to think about next generations. Yeah, and that's why integrated pest management is so important. Next. So what is integrated pest management? Basically, actually, I have one videos, but it doesn't show here. But I can just talk about this. I'm sure that you have heard about, you can still remember the fables of the tortoise and the hare competitions. Remember? Anak dengan kura-kura, pertandingan dia. Lepas tu, the hare is so confident, and his hare is very fast, right? Can very run and very confident. You're gonna win. And the tortoise is slow but steady. But eventually, kura-kura yang menang kan. So the winner is actually the tortoise. And in integrated pest management, just the same. Who are the hare? The chemical fertilizer and chemical pesticide. It's fast. You can see the effect very fast. Farmers just love it because they can see it, right? But very soon we realize that it actually do more harms to our agriculture farming, agriculture systems. And now we have seen a lot of soil which is become hardened. And even you put a lot of fertilizer, your plants doesn't just grow well. And then you have a lot of problems that the fruit set doesn't grow. You don't have a lot of fruits. Problems like this. And then you have more pests. They just love those plants that with more nitrogens, right? When you put in more baja, nitrogen, lebih banyak serangga yang serang. So all this, we can see more of that. So, well, else the tortoise is using the microbes, using the other biological components. It's slow, but it's steady. And slowly we will reach the sustainable way. So that's why I always say that integrated pest management, so can compare in such a way, is an eco-friendly approach that controlling the pest, emphasizing the techniques, its combinations, just not the biological control, but also how we use manipulate or modulations of habitat. We try to change the habitat so that it is lebih sesuai, is more suitable for certain insect or certain microbes, so it can be more beneficial. And we also use resistance varieties. I'm sure that there are lecturers here, the faculties members here are doing plant breeding, right? So this is the ways that we try to help our farmers to have using the new tools, which is genetic engineering, to produce crops that is more resistant to pests, and even now more resistant to climate change, like drought and the floods. So I think that is why integrated farming it is about. And the goal is to manage the pest populations in an economical viable, means that as long as it does not cause too much damage, we should endure certain populations there in an environmental responsible way. And just imagine that, let's say the tortoise masa dia berlari, so during the rest, now it's not just dengan persistence, but also with the new tools like the GPS and the drone, things like that. Imagine that there is more chance that we can win these things, right? So let's say we're using the integrated farming integrated pest management system, the one that we have now, plus with the new modern tools that we have. Imagine how much we can achieve. And that's why today's my topic is about redefining the integrated pest management, old wisdom, but with the new tools. Hopefully we can do much better. And next. So what is the purpose of we doing integrated pest management? You see my slides, not many interesting slides. So the reason is that so they can focus on me. Okay, the story. Okay, so the integrated pest management, just like any pest management, for us to increase the productivity, as Prof had mentioned, the populations is increased. Even with the COVID-19, we still have a lot of people on Earth, right? And we cannot just like the movies that with the step of the one finger, half of the population disappear. We don't have that kind of tools. Populations is increased, food demands is there. So can we increase the productivity? We have many ways. Last times we used biofertilizer, all those things. And then slowly we have chemical fertilizer. People put it a lot. And then they use a lot of chemical pesticide and things like this. So the productivity actually increased a lot. That's why we have industrial eras that people can give birth, more children, things like that, and the population just keep on increasing. But soon we realize that what we do is really negative. It's not only just to the agriculture, soil become less fertile, it also affects other animals in this world. I give you an example. I'm sure that you remember those DDT, racun. In 1970, those years, it's very, very popular because it's very effective. People can see it. But sooner or later, they realize that this is very harmful, not to humans, that cause more cancer problems. I have a lot of friends, very young friends. They are now suffer for cancer and die for cancer. I think the incident is getting more and more. And then we also realize that, like, I give you an example, like the eagle, the bald eagle, the population also reduced because of the DDT, because the DDT can reduce the calcium, less calcium retained in the body. So the egg, dia telur, become less calcium, become less fragile, become very fragile. So belum sempat dia menetas, before it becomes, to have to give birth to the child, to the eager birds, it becomes menjadi telur yang kita makan, what do you call that? The sunny up, the egg, because it breaks, cannot hold the eggs any longer. So we can see the effects is not only just to the humans, but also to the eagles, to the birds. When it attacks the birds, what happens to the worms inside the soil? Because this is it's a food chain, so it affects everyone. And then the pests become more, and then it attacks. So the cycle becomes worse and worse. So we need to take responsibility to stop this. And I think that is why all of us are gathered here, not only because we are academicians, and also for the future generations, our young children. You get educated. When you become a policymakers, you can make this clear that we need to protect every organisms in this world, so that we will maintain this beautiful world. I can tell for sure. I can tell for sure if we continue this, humans will disappear, but others organisms such as microbes, they will be there because they are more resilient. Even with the UV, with the nuclear, everything, they will be there. We are the only one who's going to disappear. Yeah. So try to do something. So we need to avoid the excessive use of pesticide. And the next. But however, we still humans. We still would like to have more interest for us as a humans. So if let's say I'm a farmer, I'm planting paddy, right? I won't be so generous or so noble. Okay, let the pest to enjoy the food. I take a little bit what is left. No, I would prefer to have more paddy so that you can take some, but more should be mine so that I can harvest, I can share, and I can make profit. So this is called about economic. It still have some profits. That's why we say planet, people, and profitability. You should have. If not, no farmers would like to do farming. There is less and less. If nobody is going to do farming, nobody is going to rear any things. Eventually, me and Alex will be the happiest because we're going to make a lot of money because we're going to only eat meat from microbes. We just have the land and we can produce a lot of meat, vegetable, everything from here. And you just cannot enjoy any daging yang betul, sayur yang betul. You can just only enjoy everything from the lab. We'll be happy, but is that what we want? I don't think so. Yeah, I don't think so. So we still need to manage the pest damage with the economical way. But bear in mind to reduce, if we cannot totally move away from the negative impact, to reduce the impact to the humans and the environment. And next. And then by doing this, we need to conserve biodiversity. As I mentioned, this is a food chain. Kita punya knowledge, our knowledge is not enough yet to know who can we do without, who can we do with. We still need all this. If we have more diversity, we can survive this climate change better. We have better chance. We really have a better chance. I think just now, Prof. Alex has shown one of the microbes. They have to live together. They fall together. We call it consortium, right? So they have to live together. If they are separate, they cannot live better, or they even die. Even that you have one that is more stronger, but it cannot propagate more. Just like us, if we just one self in this world, the chances that we survive is very, very much less. That's why we have to have biodiversity. Yeah. And then next, then we want to reduce, yeah, good. Then we have I think semua ialah memang buat IBM, you know that this is the basic frameworks of components. You can have 5 or 6, but actually it's the same. The first one is to do preventions and then monitoring and threshold and control and evaluations. I do one by one, the next. So next, preventions. So cara yang biasa, the old knowledge is about cultural practice, where we will use the new planting materials to resistance to the pest. I'm sure you have heard about Bacillus thuringiensis incorporated into the maize, and then it can more resistance. That is one. Indonesia sama dengan Malaysia, kita banyak kelapa sawit kan? So we have a lot of oil palm as well. Now we are going to the pest resistance and disease resistance material as well. Sini tak tahu ada Ganoderma disease? Yeah, sama. Di Malaysia pun sama. So they are looking for those, I will not say resistant, is more dia punya damage dia kurang sikit. So we said that kind of varieties, so they have more tolerance, that is the word. more tolerance towards the pest and disease varieties. So ini bukan satu ilmu yang lama. This is not a new knowledge. It is actually an old wisdom. I think the old generation, kalau dekat kampung, in the kampung, the people also started to, okay, this one actually can be more resistant and so on. That's why we have a lot of paddy varieties, right? Remember? So this is but with the new tools is what? Genetic engineering. So can we do this? But genetic engineering also have concern because of the GMO, but a lot of people are using more natural plant breeding, but it takes time. But ingat kura-kura, ingat the tortoise. Sometimes slow way, maybe it's the sure way. Yeah. Another one I think that everybody is practicing also is the crop rotation. That is what we call ancient knowledge. In Indonesia, in Malaysia, I think in Saudi Arabia, maybe years before, they also have they will open one land, planted one thing, and the next year they move to other place. And then maybe after 10 years, they come back to the same place. So this is called crop rotations. I mean, this is one way, but now we don't have the luxuries. This is government land. Ini Pak Ali punya land, and that one is somebody else's land. And we only have one small land, and every year is that land. So can we do something about this? So some people, they still practice crop rotation, means that after the harvesting, they let the farm rest for 3 bulan, 4 bulan before 3 months, 4 months before they're replanting. There is another way, and then they plant different crops. First, maybe vegetables, and the next maybe they plant the root like sweet potato, and the next they. So why do we do this? To reduce the pest pressure, because pests sometimes they prefer certain food. When you change, they will eat. Then when you change the other crops, then you will reduce the pests from building up the population. So that's why we're doing crop rotation. Another one very popular way is to do sanitation. Move out those disease plant, right? So that you can reduce this. So there is sanitations. Oil palms now. Some people, they don't practice this to move those fronds out. They don't do that so that they can maintain the organic metals there. That is also for carbon sequestration purposes. We have done study on that. Actually, if you remain there, you do proper way of management, it will not increase the pest. So not necessary that we have to remove everything out from the equations of the landscape. Yeah, so that's why research is very important. Yeah. Another one way, which is I really like to talk about this, priming your plants for sustainable agriculture. Priming maksudnya kita bagi kita punya tanaman expose kepada sesuatu penyakit dalam jumlah yang sedikit, so that dia menjadi immune kepada dia. So prime next. Okay, priming the plants is actually to let the plants to acquire the stress memory. You may think that plants have memory. Does plants have immune system? They do have. It's just that it's different from us. It's just a little bit different from us. They do. If you can recall, in 2020, every one of us have the vaccine, right? For COVID-19. Vaccine is a similar concept. Vaccine is actually that you have reduced toxic or reduced pathogens inside there, so that we exposed to this, and then our body can build up the antibody to go against it. So with this technique, plants is also to use sub-level of stress management. We give them kurang air, less water, or maybe higher heat, or maybe even expose them to certain amount of pathogens, not so much, to give them under a stress. So the plant itself because plant is not themselves, they always have microbes with them. That's why microbiologist is here. So we have microbes, especially in the root system, also on the leaf. They will communicate with the plants. And sometimes these microbes will tell the plants in terms of in their own languages, usually it's chemicals, to tell them you must express this much. So they will ask them to switch on certain genetic, like producing more salicylic acid or jasmonic acid, so that they can trigger, with this, they can have better to repel the pest better. So that is the way that we do things. That's why Alex have talked about putting microbes to make the plants grow better, more resistance to the certain pests and disease. This is same concept using the priming. So priming have two ways. Number one, using the seed. Before kita planting, sudah kita bagi dia memory untuk supaya when the next attack come in, they already know how to kickstart to the immune system. So another one way is put it into the soil, which is the rhizospheric microbiome. So, and the good things about this, some of this memory can be passed down to the next generation. So it's transgenerations, and this has been proven by science. Yeah, so I don't want to go into detail of the mechanism. There are some switches in the genetic that can be passed down. Yeah. All right. Next one. That is about prevention. Once we do a prevention, that is what the best we can do before we plant. After planting, well, the other insect, they also have their ways how to overcome the preventions, right? Well, we just want to reduce the risk. So the next one is that we keep on monitoring. So how do we do monitoring? The traditional way is to using the trap, right? We put some pheromones, so they're trapping, then we just look into see what are the pests that's present, the density, and we're also using the visual inspections. But all this, number one and number two, is really labor intensive. Kita kena jalan, tengok, like Malaysia and Indonesia, very hot, right? Yeah, I couldn't bear the sun. So you will try to do less of this, but this is very important. So what about the new way? I don't know whether, I think same is Indonesia, Malaysia, a lot of paddy area. Now the farmers is all the old folks. The new one, the young one, they prefer to do it in the office, go to the, to the industrial area with, they prefer that, better income actually. So that's why we have less. So the older farmers, how are they going to do the monitoring? Basically not. That's why we have problems where some of the good paddy, nobody is planting. So the breed just gone. It means one of those genetic is gone forever from this world. So what we can do is that with good things is that we have the new tools. We have the drone assisted, okay? They can use it in the military, but we can use it in our own field. And today, my friends, Coco is with me, and she's also an expert in the drone-assisted farming. She's looking after a lot of oil palm assets, and she's using a lot of this drone. So what does drone do? Okay, drone is just basically a fly to help us to cover more place. But then we need to have sensor. So there are many different type of sensor, and just like plants is like humans. When we are sick, you basically can tell, today you doesn't feel well. You can feel this. And the same thing with the healthy plants and the sick disease plants. There are also some symptom. So we can actually call for this one. Mungkin dia ada bintik-bintik, and maybe they have some scorching from the leaves margin. All this we can actually see. Another one is using the infrared and the blue red. The the healthy and the diseased one, they emit different visible lights. The healthy one, they have more visible lights because their photosynthesis is more. So photosynthesis, we use a lot of the blue regions and the red regions, right, for photosynthesis. So they absorb more untuk photosynthesis. So for those who are sick, they cannot do more of this. They release this. They release this more. Yeah. But for healthy plants, they don't need a lot of green. So they will push out a lot of green during the photosynthesis because there is not needed. So with that kind of knowledge, we can use the drone and the camera, a good camera, to do this, to monitor. So that is I would say, old wisdom with the new technology. Of course, we need to continue to gain more knowledge how to distinguish between the diseased one and the healthy one, right? So that is another one. So that comes to the data analysis. And data analysis is a big topic as well. So we have to stitch all those maps together, and then how do we differentiate? So there are called normalizations of differential vegetation index untuk memberi perbezaan antara tanah yang banyak air dengan tanah yang tidak banyak air. So these are things that now we already have, that you just fly, you know that this patch of soil is more moist and that is less moist, and this is more having more maybe ions and that one have less. So these are the tools that we have, but we need to continue to improve from here, and that's why the young one are here. Okay, next one. And I think the threshold is the key to the success, because previously, the farmers, what they do, a lot of farmers, they want to take an easy way out. I just keep spraying fertilizer. I just keep on spraying pesticide. My plants will be very happy and I will have good yield. But they don't know this will going to harm the soil. So now we, the IPM, we really need to have economic injury level for each of the crops, important crop. So we know that when do we need to spray? How much do we need to spray? Yeah. And then, of course, spraying, we can use microbes, and then we can also use, I'll come to the control later. So this is the threshold. So whenever I do my research for any crops, first, we will look at what are the pests, what are the disease. Second is, what is the economic injury threshold? Bila kita nak ambil tindakan. Next, then we will look into what are the control methods. There are 1,000 types of control methods. You know, there are chemicals, there are different types. There are, you can just a mechanical way, physical way. You can have chemical way, biological way. So I'll talk one by one. As I mentioned, there is not much pictures, so you basically have to just listen to me. The next one. So we talk about the first control is to boost up the plant immune system, just like bagi vaccine. So how do we do this? We're using biofertilizer, we're using biochar, soil amendment agents, mainly type of soil amendment agents. Actually, had been covered by Alex just now. A lot of this. Mungkin Papa, Ibu ada banyak juga buat eksperimen tentang biofertilizer, biochar. So the concept is using the microbes as well as the planting material, the biochar itself, to boost up the immune system. Just to share to those students, young students, maybe you're not aware, the plant have their own immune systems. It's not like similar to us, but they do communicate. So I give you an example. Let's say I'm sick, I have been attacked. I do have an immune response because the microbes will actually create a pattern in my body, and my body will recognize, oh, this is a microbial associated patterns. So when I have this one, it will trigger the other system to produce chemical, the biochemical, which is like there are many types. Just the popular one is salicylic acid, jasminic acid, and so on, to protect. And also, this not only just to protect, they sometimes to make the root menjadi lebih keras, thicken the cell wall. So in making it thicken the cell wall, so these are the mechanisms they use so they can improve the immune system. So sometimes they will produce certain smell of chemicals that become not so much liking of the other pests, insect, so they will repel. So that is one way of doing things. Another one is that, let's say I'm being attacked and I have microbes that associated with me. So I was sending some message, dan microbe itu akan enhance the message, send to the neighbor plants. So plant yang di sebelahnya akan tahu, okay, ada kawasan sini ada serangan sesuatu. So they will started to take precautions, which is they started to have some response. So that is the amazing things. Just like what has been presented by Prof. Kita punya badan banyak microbes yang membantu kita dapat nutrients to get us a lot of nutrients. Same thing with the plants. They need microbes to help them to absorb nutrients. Therefore, who runs this world? Actually, it's the microbes. The one that runs this world is actually the microbes. But humans, we are given the intelligence by the God. Yeah, the God gave us the intelligence so that we can use some of the microbes to our interest. Yeah. Okay, next. The next control methods is bioinsecticide. Popular one, just as mentioned by Prof. Bacillus thuringiensis, Bacillus. I have some friends from from University Brawijaya. They're doing on the Bavaria. These are all the bioinsecticides that you can use. Not only from the bacteria, sometimes it's from the fungus. So I'm also using this one to control. I'm working with the oil palm also on the fruit orchards, and also working on Sarawak pepper plants. So we have used this. We have shown this is very effective. Yeah, because tadi saya ada kata root knot nematodes. Menyebabkan kita punya tanaman punya roots itu tersimbul, and that we also using some of these fungus things to help to fungus yang boleh serang nematodes. And I also use some of the biocompound like salicylic acid, and it has shown that the nematodes incidence rate drop down a lot. Yeah, because it's under the ground, some farmers cannot see it. So bila berlaku semua, it just sekelip mata, mati semua. So we have to take precautions with this one. So this is another one method, biofertilizer insecticide. Next one. Next. I think that this pheromones is very popular. Oil, neem oil, pernah dengar neem oil? Neem oil has been used quite extensively. A plant extract, because all these actually can improve the plant response to this. Yeah, sometimes we use these pheromones to also reduce the pests. A lot of this, like neem oil, is to mengganggu, to interrupt the life cycle so that they cannot breed. So we reduce the pests. Yeah, we don't kill them. A lot of people use neem oil. They say it's not effective. The pest does not die. It's not the focus is not to make the pest die. It's to stop the reproduction. So therefore, it reduces the pest. Yeah. Okay. Next. So another one. This one, as Bacillus thuringiensis, everyone know that this involves the external genomic insertions. So this is involves genetic engineering. So a lot of people will be cautious. This is GMO, but I'm not sure whether you know, every one of us at one point of time that we actually use GMO things. You just check your shampoo, they may have GMO content things. It's something that you does not run away from. Malaysia, we don't have the legal, we do not put it so stringently that every label you have to write GMO. If you know that, you know that actually most of the product contains certain amount of GMO, right? But BT has been around for centuries. but there are other microbes that we can make use of. I'm sure one day we're gonna use the one that from from the volcanoes and from the Red Sea, things like that, that can and also from the dark soil. Yeah, we also do it like myself. We have a lot of projects working on the soil from fertile soil as well, and we believe we should use the microbes that from the country, from the locality, so it becomes adapt better. So usually we will look for those, and we look at we characterize their function, what is their functions, and then we incorporate it into this. Next one. Mine is very short. And then semiochemicals, spelling says semiochemicals. This is also now very popular, has been used. Basically is also to improve the plant immune system. It is, I'm not sure, when people talk about organic, they thought that is totally no chemical, but we actually build up by chemical. So semiochemical is basically substances induced by this plant or the pest that actually carrying certain signal to the plants to do certain response. Yeah, it's a big topic. Maybe I don't want to go into detail, but basically there is a group of these substances. Yeah. Next. So therefore, there is so many control methods, yeah. And then after that, we do evaluations. When we do evaluations, we're not only just looking into how the crop performs, we're also looking at how the soil performs and how it affects the environment. So we consider the external and internal. I think one of your colleagues here, Pak is going to look into this. So we have experimental plots. We're using different microbes to amend the soil, and then we also do intercropping. We have different types. Intercropping is very interesting because every species of crop, they actually attract different types of microbes. So can we use this intercropping? Then you have all this, we can actually examples a stronger defense system for the crops, and also reducing the pest. Yeah, so we're going to do evaluations. And we have a few experimental plots that have very, very good effects we have seen. 2 days ago, one of the farmers, the crop farmers for the fruit orchards, he was experimenting our soil microbe using the microbes agents with another one from the commercial one. So he planted the banana, masa yang sama, applied to one plot is with the soil microbial treatment, the other one using the one from the commercial one. So at the first 4 weeks, the 3 weeks, he see the growth from the commercial one grow very, very fast. And the other one looks like, okay only. But after 3 weeks and 4 weeks, he started to see the difference. The other one started to pick up. The reason is microbes need time to adapt and need time to associate with the plants. So therefore, the 3 weeks, we see that is low. But these farmers is very persistent to want to see. After the week number 5 and 6, he started to see that the growth about the same. And after that, the other one, boom, faster. And without any fertilizer, just purely microbial. And the microbial, what they do is that the microbes that we use is basically to help to fix the nitrogen, which is already in the environment. Another one is the phosphorus and potassium already in the soil. It's just that it is in the form that the plants cannot take. So the microbes help to transform them into a form that can be absorbed by the plants. And without adding any additional fertilizer, we actually make the soil more fertile for the plants, you can see. Yeah. So this is some sharing by the farmers that I would like to share. I'm very excited as well. Next week, I'm going to visit them to really record down whatever they said and to measure scientifically way. Although we have a lot of data back in our experimental plot, but this one is in a big scale. Yeah. So next, with this, I would like to say thank you, and all of us are important with the net. We're going to make this world difference. Let's work together to cultivate a future where our planet thrives and every one of us can enjoy a nourishing and sustainable food resources and the next generations to come as well. So with this, I really thank you, and I hope that we can form a network here, all of us, and just like you, a network is important so we can each come up with something to help this society and this earth that we have. Thank you so much. Thank you.
All right, thank you so much for Prof. King for that wonderful presentation about integrated pest management, the slow but steady approach for sustainable agriculture. Now we go to the discussion session. Is there any question for the participant in here? Okay, maybe we have a question from the participant online. Okay, one question from Nur Edi from Tadulako University. Dear Professor, I am Nur Edi from Tadulako University. Thank you for the very interesting topic presented. I'd like to know about the microbes used for bioremediation in polluted mangroves. Would you like to explain more related about the microbe use and how to apply? And do you have any experience about bioremediation, example nickel mining area using microbes? Warm regard from Palu, Central Sulawesi, Indonesia. So I think this is for Prof. Alex, right? Okay.
Yes, thank you, Noor, for the question. We applied this bioremediation protocol a few years ago in a very heavily polluted mangrove. And the idea in that time was to combine those microbes, as I explained. I mean, some microbes that were able to degrade different fractions of the oil. Because remember, the oil, the petroleum, the oil, it's not one oil. It's also diversity of chemical compounds products. So we don't have a super microbe that can degrade every different fraction of the oil. And actually, we isolated one that was able to degrade more than 90% of the oil, but it was a very slow-growing microbe, so it was not good for the application in the field. So it's interesting in fundamental research, but not for the application. And for the application, we select some particular microbes, and the best consortium was composed by 10 microbes, different bacteria in that time. After that work, we also add some fungi, and remember the mangroves, we also have the anaerobic conditions. And in the first consortium, we only applied aerobic microbes. Some of them were like belonging to Pseudomonas fluorescens complex. Pseudomonas are known to be able to degrade different fractions of oil. But actually taxonomy in microbiology is always changing. So this is why it's difficult to, because in that time it was a Pseudomonas sp, one of them. That one was a Penibacillus sp, some actinomycetes as well. And some, some maybe known famous microbes that we are always using. But actually, after the use of the new tools that we have now, the taxonomy is changing, and some of the microbes that we used in that time, now we know that there are new species as well. So some of them we actually still naming. Yeah, but I mean, the good thing was that, well, worked. And we, of course, did some tests to test if they were antagonists to each other, not like normal things that we do for agriculture or something like that. And the big issue was in the mangrove was the tide problem, the issue. So when I applied the microbes, the tide come and wash away everything. So we applied a normal thing, like we have the chemical fertilizers that we can have slow release. So we did the same approach using alginate in that time to contain the microbes. So it was like a protective, way to preserve the microbes. And we did some shelf life experiments, and the microbes were actually alive inside the polymers of the alginate for up to 6 months in the refrigerator, so we could keep them alive. And when needed, we just applied the mangrove. So with the alginate encapsulation, we dealt with the issue of the tide. So when the tide came, there was a swallowing of the alginate and slow releasing the microbes. So they performed the effects in the mangroves. And when the tide went out, the alginate shrank and also protected the microbes inside the alginate from the sun, the radiation, the drought, and because this was a Brazilian mangrove in the northeast, so close to the equator line. So it was always super hot over there. And so that was really an interesting approach. And I forgot the rest of the question. Yeah, but yeah, I think that was the main thing. Now we are testing some cheaper polymers because alginate is very powerful. It's been used for a while. But since now we are talking about huge, fields, hectares of kilometers, actually, millions of plants. And so it's we really need more cheaper and reliable polymer. So we are testing some we are teaming up with some colleagues from the chemistry and polymers laboratory. So testing some, eco-friendly, also polymers, but with this is low release capability and cheaper to produce. Let's see the next steps. Yeah. Thank you. Thank you.
Thank you so much, Prof. Alex. And then, is there any question? Oh, okay. Pak Lukman. Saya mau bilang monggo ini, Pak. Okay, Mr. Lukman, time is yours.
Thank you. Very interesting, both of your presentation. And I have several questions. First for Professor Rosado. So very interesting about microbiome. And also we are here starting to study about microbiome by metagenomic, but still we face some difficulties in how to analyze, and also maybe in the future we can collaborate.
Yeah, sure. That's idea. Yeah. Yeah. Yeah.
Yeah. And I have a question about, that metagenomic and also for microbiome, this is a holistic approach here to analyze the whole biota, especially for microbe in the habitat. But after that, in the end, but usually we use only 1 or 2, 3 microbes only, because sometimes in the nature there is a very complex microbe that has rule, and they work together. But when we study, in the end we only 1 or 2, something like that. My question is how to apply this whole holistic approach applied in the field, particularly in the agriculture context. And one question also for Professor King. Nice to meet you also. And you mentioned about the prevention approach about transgeneration memory and the sublethal stress-mediated reprogramming. I'm still do not know about this concept, or maybe any example about how this applied in the IPM. Thank you.
Okay, yeah. Yeah, so the idea is really to collaborate. I think this is why we are talking about this. I mean, it's science is collaboration. There is no other way. And we have different expertises, and we must combine our expertise towards a better and sustainable environment. There is no other way. I mean, this is period. And yeah, I'm totally open to collaborations. We've been doing a lot of omics research, and I'm not bioinformatician myself, but I know the basics, very basic. I know how to turn on the computer, but I'm kidding. But now I have bioinformaticians in my team, because as I mentioned before, the field is changing so fast that we can't cope with the new tools, the new softwares, new pipelines. So we really need the bioinformaticians as well to combine. But I agree with you. I mean, the question is a $1 million questions. I mean, yeah, a question: how can we leverage the power of the microbiome itself? Because usually we rely on one or two or a few isolates, and that's the thing. And they are performing really well in most of the cases. We have very good examples of. We have also in our lab in Brazil, for instance. I have a colleague, Mariangela Hungria. She's a Johanna Doberainen was a pioneer, but Mariangela is leading the field now in soybean in Brazil. And in Brazil, we don't use actually chemical fertilizers for soy like in US. They rely on a very heavily use of chemical fertilizers. In Brazil, it's only biological nitrogen fixation. So using the selection of the best strains and the best combination of strains. So that's really reliable. And this is going well, but I think we will need more. We are still not there yet, but I think this is the way that we are following now, how to really rely not only one or two microbes, but how can we really, and this is actually a new open field, because now we have the tools to understand what the microbes are talking to each other, like using metabolomics. And this is valid for human gut. So there are a lot of new improvements and treatments now being tested now, using not only one or two strains for probiotics, but also how the combination of those probiotics, for instance, or using also the fecal transplantation, they can change the whole microbiome. And how to maintain this microbiome in a steady state that can really perform, and not to need, not adding always the probiotics to keep this going. So it's a lot of challenges. But as I was saying, I mean, I don't think we are there yet. I mean, but we are almost there. And with the tools that we have now, we can really try now to answer those questions. Because as I mentioned in the beginning, was just a debate for the academics. So this can be possible, or this must be possible someday. But now I think we have tools that we can really, push the boundary to another level. And this is happening now in the medicine, in human microbiome advances, and soil microbiome as well. And I think in the next few years, we will see a lot of those changes and those products in the market as well. So it's really a movement, a very strong movement now towards this direction. And thank you.
Thank you. Thank you for your questions, Pak Luman. I think this is the second time I meet you online. Yeah. So actually, I like the questions that you posed to Alex. I think Prof. Alex and myself, we believe in cocktail. Yeah. We call it consortium. I seldom apply to single strand. For whenever I do soil amendment agents, definitely it's a cocktail, because each microbes, as mentioned by Prof. Alex, no matter what we are doing, we are dealing with the biological materials. They have many different components that we need to, let's say, want to hydrolyze it, we have to cut down different components, and different microbes actually have different enzymes that cut different, even though they can have multiple enzymes that work on different angles, but sometimes they still need to other microbes to support them in other categories. Yeah. So and actually I just wanted for discussions purposes. This like we're working on the different soil, poor soil and compare with the rich soil. And when we do a metagenomics, we will see that they from what we have studied, always there will be a group is called microbiome. It always there, no matter it's a good soil or poor soil. So that's why I say it's important to have that kind of tools for us to know that this is the main groups there. But what we want interested is actually the minor one that is functional. And once we know what are those, is always present in the good one. So these are the group of microbes that we would like to characterize more and then put in not just single, but as a cocktail. Yeah, I think that is the approach now. Yeah. So questions by you about the transgenerations memories. Until now, the scientists had not yet finalized, and we don't know what are the underlying mechanism. But when we do study it on the molecular level, we notice that some plants, after they has been exposed to certain sublevel of stress, there are some changes in the molecular. Remember the DNA on the histone things? So the histone can be methylated and the DNA can be methylated and can have some other transformation that change the expression, the gene expressions. When they change the gene expression, certain gene expression has been up-regulated, some are down-regulated. And therefore, they can respond to the stress. And don't know somehow, this kind of modification, somehow it actually bring down to the next offspring. When the mother plants have produced the next seeds and so on. So, but it's not always, sometimes the changes is somatic. It does not bring down to the next generation, but some they just bring down to the next generations. And for us, we just know that there are some change modifications on the molecular way, but we don't know how it actually triggered this. So just like what Alex said, we are excited because we have more questions to answer, but we don't have the answer of all. We have more questions. I think that's why R&D is always keep on evolving, and then we know more. Thank you for your good question. Thank you. Thank you.
Okay, thank you so much, Prof. Alex and Prof. Kim for the answer to Mr. Lukman. And one last question maybe? Okay, Bu Karinda. Thank you, Bu Yohana. Okay, my question to Prof. Alex. As far as I know that Bacillus thuringiensis is an entomopathogen pathogen. But you have mentioned that in your example that BT become an endophyte. So my question is the BT have been engineered?
Yeah, that's a good question. Not really. That one, not. That strain, the new strain that Isabella isolated. We are studying now the genome of that bacteria, in deep, to try to understand the effects. And actually, Patricia raised a very important question because we still don't know, how the microbes can influence the epigenetics of the plants. Or, we've been seeing this also in some other studies in my lab in collaboration with some colleagues, that the microbes, the microbiome can change. You can, change the epigenetics, or so the genomics of the host in corals. This is quite new. So the same for plants and the same for humans. And we are still trying to figure out what is really happening. I hope to answer your question soon. Isabella is working really hard in the lab now, trying to understand, what, because she was just trying some different combination of novel strains. And she found out that that particular isolate, those actual isolates, also the other one, the Penibacillus brasiliensis, and the other one, polymyx as well, they were quite effective, in the expression. She was trying several different things. Actually, because the first idea of her thesis was not working. So we came up with a plan B, plan C, and something like that. So this is something very interesting happening here. And now, we are trying to figure out what is really happening, because the effect, and especially the combination, it's not only one or the two, but a combination of those microbes. It's really helping, including the tomatoes when she's trying to grow in very arid soil from Saudi Arabia. And we can see the difference between the control, it's huge. And also she compared with chemical fertilizers as well. Of course, chemical fertilizers give a bust, but the strains without chemical fertilizer also start a slow process, cura, and I like the concept, and now we can see, catching things. So it's something interesting happening there. Let's see next.
Yeah, I assume that this bacteria become a vegetarian.
Very good. Maybe, yeah.
Okay, for Prof. King, that you said that using the drone, but I think I know that some farmer using drone to spray the pesticide. Yeah. So is there any survey or research that where is that I mean the safety, the safety for the using drone to spray like that?
Very, very good questions. Actually, using this technology is, I would say, it's still quite early in Malaysia and Indonesia. There is a regulations for flying drones. There are certain zones that we cannot fly. I think everybody knows, especially near the airport and things like that, they cannot. But using the drone to spray pesticide, right? There is still a lot of research because different area, the wind, the strength also can drift. There are problems that the certain chemicals or certain fertilizer has been drifted because of the wind. And so how low it have to come down. This is all this, and then the nozzle, the droplets, the size, all these are we have to study. So the delivery of this is a new technology. We are excited. But I would say that they still have a lot of challenges. Still, whenever we go to the farmers, when we talk about drones, we will tell them we need to optimize. Setiap every farms they will have different. We have to optimize because of the terrains is different, and then the crops are different at different stage. How far that we are, and also certain pesticide, we cannot just spray on that. We have to figure out another delivery system. Yeah. So a lot of challenges there. Yeah. It's excited. We're very exciting about this, but then challenges are there. Yeah.
Yeah, I think if we want to spray BPH, brown plant hopper, I think it's not effective, I think, with the drone, because the brown plant hopper, the cure is in the stem, so in the near the soil. Yes, it's flying is not good, but here for the paddy, brown plant hopper is the key pest.
The Sembora. Yes, that's why I said the delivery system is very important. Certain like those borer, stem borer yang masuk dalam padan untuk penggorek itu susah gunakan spray. So it is only a method that one of the methods that we can think about. But the delivery system is always innovating. Like how do we deliver this one with less humans? And then you can use the drone with if let's say you have to inject, we have to use a drone with a camera to tengok dekat mana dan dia buat suntikan. So this is a lot of technology. We have to work with the company to design this so that we can actually have the better delivery system. That's why it's a lot of optimizations. I truly agree with you. Some of the disease that happening in the stem as well as the root, it's not that easy to use the spraying. Spraying is the for those Lepidoptera insect that is easier because of the contact, you can already have the effect, but some there is no effect, like the ana termites, you can't use spraying. You have to use betting system and so on. Yes.
Yeah, maybe it will be more effective for kelapa sawit.
Sebenarnya kelapa sawit pun because kelapa sawit a lot of pests and disease like Teradaba mandela, the bagworms, very effective using the spraying. Very dan mereka tak gunakan organic pun, mereka gunakan banyak chemical juga. So they use a lot of chemical and that is effective because kelapa sawit biasanya beribu-ribu hektar. So for them spraying is a better option. That's why I say it depends on the pest and disease nature. If it's those inside the plant, we can't use spraying, but those outside, yes, we can. And those we can also spray on the soil. Yes, China have used a lot on the spraying on the soil. It can be effective, especially on microbes. They can help to modulate. But the effect you have to see for a long, we have to wait and see. Yeah, yeah. So thank you very much for your questions. Thank you.
Okay, we still have a lot of questions because we have more than 100 participants online. But I'm sorry, because lack of time, we have to move on to another session. If you have additional question for the presenter, you can send email, maybe to Prof. King and Prof. Alex. Thank you for all the presenters today for sharing your wonderful presentation, and thank you for attending this session. All right, I'm giving back to Bu Hafidah. Thank you very much, Prof. Alex and Prof. King, for the enlightening presentations. And thank you for the moderator for leading the engaging discussions. And we will now take a short break until 11:05. So we will have around 10 minutes for perhaps relaxing or stretching your body, or perhaps you can go to the restroom for a while. And please come back we will be back, or we will start the second sessions at 11:05. Jam 11 lebih 5. So for the participants, for the online participants, we will reconvene at 11:05 EM. Thank you so much.
Tina, are there any jobs for stay-at-home moms that can be done while still taking care of their child? Of course, virtual assistants. So, as a
Check, check. Okay, check. Check, check. Cek, cek, cek. Halo? Sudah masuk suaranya, Mas? Halo? Oke. Sudah ya. Oke. Oke, cek. Okay, our short break is already over, and we're going to continue to our second plenary sessions with Prof. Agus and with Assistant Professor Dr. Suradet Butakon. So for all of the participants, please be seated. Okay, good morning, ladies and gentlemen. So welcome back, everyone. And our next parallel sessions will features Professor Dr. Agriculture of Science, Agus Tarno from Faculty of Agriculture, Universitas Brawijaya, and Assistant Professor Dr. Suradet Butakon from Kasetsart University, Thailand. They will share their expertise on the latest plant pest and disease management advancements. But before that, let us check whether Prof. Suradet are on Zoom or not? It's already on Zoom. Let us check first because I want to greet him actually. Good morning, Prof. Suradet.
Yeah, good morning. Can you hear my notes? Yeah, I can hear well and everything is okay. So
Very much for coming. So we are happy that us this actually in Indonesia. Okay, thank you so much. And the sessions will be moderated by Ms. Fralita. She is a lecturer in the Department of Plant Pest and Disease. And please welcome for our moderators and speakers. Give a big hand, big applause. Okay, for the rest of the discussion time, I will hand it over to Ms. Fralita. Discussion. Thank you very much.
Okay, thank you, Master of Ceremony. Good morning, everyone.
Morning.
Okay. Yes, in this second session, we have two speakers that are joining us with virtual and also in here, Professor Agus Tarno. Okay, before we start the presentation, let me introduce you who's the speaker that will give the views in this second session. Okay, first is Professor Dr. Agriculture Science Agus Tarno SP MP, and he is a lecturer at Department of Plant Pest Disease, Agriculture Faculty, University Brawijaya, and he finishes his doctoral degree at Kyoto University. He concentrates on plant protection, environmental science, and of course entomology. And now Prof. Agus is head of double degree program in Universitas Brawijaya. So you guys, if you you are attracted to join double degree, yes, we are with him now. Okay, and our second speaker, we also have amazing speaker from Thailand. Let me say hello first to him. Good morning, Dr. Suradet Butakon.
good morning.
Okay, good morning. Okay, Dr. Butakon is affiliated from Department of Entomology, Kasetsart University, Thailand. His skill and expertise are about natural product and also entomology. We have two entomology expertise here, so maybe we will talk about more about entomology here. So we have two expertise in this session, and for my information, Dr. Suradet Butakon is also an assistant researcher in CIIMER at Department of Chemistry, University of Porto, Portugal on 2016 and 2018. A guest speaker will share his views for around 40 minutes and then followed by Q&A session. So please be informed that after your keynote speech, the speaker Participants can ask the question directly on the site, or those who are joining on virtual can ask on chat box. Write your name and question. Okay, now let's join us in welcoming our first speaker, Prof. Agus Tarno, please, to give your keynote.
Okay. Thank you so much. This is first time for me to be speaker in my university. I don't know why, maybe because of there are several professors from other university, from foreign university. So I have to accompany them. So not because of I'm keynote speaker, but I have to be accompanied a speaker. Okay, previously we have microbiology session, and now we move to the entomology. Of course, maybe we can also discuss about microbiology related to the entomology. In case of me, I also have experiences on the nematology. In our department, entomology belong to the entomology, but mostly nematology working closely to the plant pathology. So it's quite difficult for us to determine indeed precisely about the nematology. So maybe that one is one of the important, one of the benefit for me, because I can involve in the entomology society, and also I can also join in a plant pathology society. Yeah. Okay. Let's start presentations. Okay. Okay, the first slide, please. Yes, this is the title that I want to introduce to all of you, because I'm working closely to the entomology. So in this session, I want to focus on arthropod community. However, actually I'm focused on ambrosia beetle, but now I have to focus on the general entomology. As in Indonesia, we have very unique apples. However, if we want to compare with another apples in another countries, mostly our apple is quite small. Yeah, so maybe because of the variety of our apple is different compared to original countries that have the apples. Yeah, we have a crab apples. Actually, the species of our apple is Malus sylvestris. So this our title is about arthropod community on the two different habitat management in crab apple orchid, Malus sylvestris. Okay, next. Okay, next. Yeah, in Malang, in Indonesia, mostly we have several crab apple varieties, and this is the 5 varieties that commonly planted in Indonesia. I have a doctoral student, they working closely to the apples, but I didn't ask him about the apples actually. But I got this one, I got information shortly from the article that mentioned about the apple varieties in Indonesia. There are several varieties in Indonesia. The first one is Malagi. This is one of the most popular varieties in Indonesia. And then we also have Anna. Also, we have a Rome Beauty, John Gold, and Princess Noble 2 Extra. Crab apple actually is not native in Indonesia, but actually native to the Central Europe, because during Netherlands government in that period, and after that apple become very popular among Indonesian people, and then we finally we imported varieties, and then we planted that kind of apples. And not so many area in Indonesia that they're suitable for the apples. Malang is the most suitable area that commonly chosen by the farmers to plant apples. Okay, next. This is the apples, but this kind of apple cannot be eaten by us. Yeah, because this apple is made by stone. So if we want to climb up, if we go to the Batu area in the highlands, actually Malang also is highland, but we can also climb up to there in the Batu. Maybe we can find apple, and then we can find this kind of apple. The 3 big apples that we cannot eat. That. Okay, continue. Okay, next. Okay, next. This is the conventional cropping system that commonly adopted by farmers in Indonesia. In commercial cropping system in apple orchard involve intensive management. In example, we can mostly easily to be seen in our area. The farmer provide a very huge volume of pesticide, and also herbicide. Another things also fertilizers. However, mostly now we also almost the same with the Brazil, as you mentioned before, Prof. We also try to change from the chemical fertilizer to be the organic one. Yeah. Okay. Next. Next. As a result, from many kind of a pattern of conventional crop system, as a result, their management can harm to the environment because they mostly use the pesticide, herbicide, fertilizer, etc. And that kind of things also contribute to reduce the biodiversity in the orchard. Okay, next. Because I'm entomologist, so I just try to bring to the arthropod roles, yeah. Actually, also important if we also try to expose about microbiological rules, yeah. But we didn't improve that one. Just focus on arthropod. Arthropod actually is a very important component in ecosystems, and they can contribute to the ecological functioning and also related to the main biodiversity component in agro-ecosystem. Okay, continue. Next. Another roles of arthropod is arthropod also provide ecosystem services. Commonly we can see from our ecosystem, they provide not only pest regulations, but also they also provide pollination and also organic matter recycling, etc. And all of kind of ecosystem services very close to the crop productions and also the soil fertility. Okay, next. Related to the ecosystem services and cultivation system that adopted by the farmers, we know that if we want to bring our agro-ecosystem to be healthy, so we have to think about how we can design our agro-ecosystem. In the designing of agro-ecosystem, we can also have to think about several activities related to the cultivations pattern that conducted by farmers. In example, there are several farmers also focus on how they can reduce the tillage. In case of apple orchard, actually not so many tillages conducted by the farmers. They just because the age of the apple is very long, different if we want to compare with paddy, etc. So in case of apple, it's very few villages conducted by them. And other things also, several farmers also focus on how they can maintain the soil cover. Now things also they can focus on how they can add the organic matter on their land, and then how they can also manage the nutrient that commonly they have to manage related to how they can support the growing of the plant. Another things also, there are several activities related to the crop diversity, rotation, cover crop, and also plant breedings. But in case of apples, I don't know very well about this kind of activities, because my friend in plant breeding didn't mention about the apples. We just use the apple that commonly appear in Indonesia. And not so many varieties that are chosen by our farmers. Those kind of activities related to designing agro-ecosystem can also support the below ground habitat management and also above ground habitat management. And if we can take a look on the below ground habitat management and also above ground management, we can have two things about how we can improve our soil health, and this one also related to the crop health. If we focus on how to build the healthy of the soil and also the crop health, we can also make agriculture become health. So this is this concept that provided by Artieri and friends in 2005. Related to that, in the management practices, we can also identify what kind of activities related to management practices can increase and can stimulate biodiversity in agro-ecosystem. Because the biodiversity in agro-ecosystem, as we know, is very essential for mediating the negative impact of intensive agriculture. Okay, next. Next. Okay, this is one example related to the management practices on the apple orchard. This is related to the ground cover. Many type of ground cover adopted by the farmers in apple orchard. Okay, next. This one also another, yeah, when we planted the ground cover, also we can also invite, in example, chicken to be involved as the pest regulation, pest regulations, and etc. Many things related to the biodiversity also can stimulate as a biodiversity around the agro-ecosystem. Okay, next. Yeah, the selection of vegetation. In here, we just focus on vegetation management, selection of vegetation management. We identified two vegetation type that mostly adopted by farmers in the Batu. Okay, next. Yeah, this is our research objective. We want to compare abundance and diversity of arthropod between the grass ground cover and also intercropping using hydrangea in apple orchard. We just to focus on how we can assess the impact of arthropod functional group to provide the ecosystem services on the apple orchard. Okay, next. Okay, next. This is our research area. We have four for each, and then in total we have 8 plot. Yeah, in each it means hydrangea, and G it means grass. So we have 4 for hydrangea, and also we have 4 for grass ground cover. So all of plot is located in Bumiaji sub-district in Batu City. Okay, next. Oh, sorry. Come back for the yes. In the right side, in the right side, we can see there are 3 traps. Yeah, there are 3 traps that we can use as the tools to identify the abundance and also richness of the arthropod. Yeah, in the apple orchards. Okay, next. So finally, we choose only 2 vegetational management in apple orchard. The first one is A. It's just apple orchard with the grass ground cover. And then the second one, apple orchard with huge planted hydrangea in the cropping there, because in the Batu also the farmer didn't only take the apple, but also they also try to take advantage from the hydrangea, because hydrangea also can be sold by the farmers as additional value. The crab apple orchard are mostly grassy ground cover with the native vegetation Cyperus leptundus and also planted with the ornamental plants such as the hydrangea. Okay, next. This is a design. Yeah, we have four for each. Hydrangea 4, and then grass ground cover we also have a 4. Okay, next. So we put several. There are several. We have a blue, and also we have a black, and also we have the yellow. It close to is related to the trap that we set up on the plot. Okay, next. Okay, next is commonly this data analysis commonly adopted by us ecologists. Mostly we use this one. We didn't have any tools in the field. We just use such kind of a tool related to trap, etc. But mostly we focus on how we can manage our data and how we can analyze our data to be more easily to be interpreted and also easily to provide the good information related to the information that collected from the field. Okay, next. Yeah, next is, yeah. Finally, we got, during our research, we short research from, it's about 4 months. We have found several classes and also orders. However, we have many, actually we have many individual and also we have many species, but we didn't identify in detail for each species. But based on the classes and order we distinguish how's different between hydrangea and also grass. And from here we can see that is about 6 classes: Insecta, Malacostraca, Arachnida, Celopoda, Diplopoda, and also Entognata. We found in both habitat managements. And in addition, also we have about more than 155 individual, and also in total we have 164 families with the 23 orders. Okay, next. Then focus on hydrangea. We have in total 55,000 individual arthropod, and that all of that arthropod belong to the 134 families and 21 orders. In case of grass ground cover, we have twice more, almost twice more higher, with 1,000 more individuals that belong to the 97 families and 70 orders. When we compare between both, is of course it's different between both. Okay, next. Yeah, this one is number of total families and total number of individuals corresponding to the arthropod order. Is focused just on the order of arthropod. In Diptera and Collembola are the most abundant orders, with the total individual about 25,000 individual, with 45% more, and 43,000 individual. And in case of hydrangea, grass ground cover, we have 42% more. And the most dominant families of arthropod actually is Psychodidae, and in hydrangea intercropping. But in case of grass ground cover, different is Cypoderidae, with the total individual is completely different between both. Okay, next. When we do analysis using this alpha diversity focus on Shannon-Weiner and also Simpson dominance, we have based on diversity in this, both habitat management show the greater diversity if we use the Shannon-Weiner and also some Simpson dominance diversity in this. Okay, next. Now, when we compare between both, we can see here is about in case of A, is abundance of arthropod in crab apple with the different habitat. With the yellow, I mean with the green one is a grass with the Fiorentina color. Is Italian football team, one of Italian football team. Yeah. Hydrangea, when we compare is between both is not so different. But when we choose based on the richness family level of arthropod, we found differences between grass and hydrangea. Here we can see that abundance of arthropod indicate no difference. But in case of family richness arthropod, we have a differences between hydrangea and grass. Okay, next. And when we try to look using NMDS priorities, we found actually still overlap between both grass and also hydrangea. But when we check, also it means based on the Permanova, it means still there are significantly different between habitat. Okay, next. Let's continue to see using the functional composition of arthropod. Okay, next. Here we can see that in case of hydrangea, based on the functional rule, there are 1, 2, 3, 4, 5. Yeah, there are 5 functional rule. The first one is the blue one is natural enemies. The second one is pollinator, and then with yellow one, and then etc. etc. As we can see, in case of pollinator, hydrangea provide the higher percentage compared to the grass. So we can see here that hydrangea undercropping was grouped into D34, herbivore, omnivore, and pollinator, and also natural enemies. 39.71% higher. Okay, next. And in case of the grass concover, in case D34s, mostly we found on the grass concover. So between both habitat management, grass and hydrangea stimulate the percentage of the pollinator higher. But in case of the grass concover, they can stimulate the percentage of the T4 higher compared to the hydrangea. Okay, next. In total, the most abundant D34, herbivore, omnivore, and also pollinator, and also natural enemy families in the hydrangea intercroppings. We can see in case of D34, Entomobridae is the most abundant on D34. In case of herbivore, Tepritidae is mostly abundant, and omnivore is belong to the Formicidae, also the most abundant. And one more about natural enemies, we have still we have remain pollinator. We have Psychodidae and also Trichogrammatidae is belong to the natural enemies family. Okay, next. Another thing related to the grouping based on the functional group is when we compare between grass concover and also hydrangea, based on abundance of arthropod, we can see that the herbivore and also pollinator greater have differences between grass concover and hydrangea. Yeah, grass concover, so the greater abundance of herbivores than hydrangea. Okay, next, in case of the hydrangea, they have the highest pollinator. When we try to check based on the richness in the family levels, we can see that the D34, omnivore, pollinator, and natural enemies, we can see all of that kind of groups is different between hydrangea and also grass. based on the family level, richness of family levels. It's only herbivore between grass and hydrangea, we didn't find differences. Okay, next. Okay, conclusion next. Yes, we just make summarize based on it's very complicated maybe for microbiology scientists maybe is quite different, yeah. But we want to summarize in details, yeah. Actually, based on our research, we can just make conclusion related to the how we can conserve natural enemy, and it will be related to the potential pest suppressions. It related to the hydrangea. This hydrangea intercropping can provide benefit because it can stimulate the natural enemies, pollinator. However, number of detritivore is not so huge compared to the grass, but also hydrangea also can stimulate the detritivore, and of course omnivore also can be stimulated by hydrangea. It's related to providing the food, shelter, and also favorable microclimate that can be facilitated by the hydrangea intercroppings. Okay, next. Another summarize also related to the, if we want to take a look on the apple orchard, farmers tend to reduce the application of pesticide. Then this one also contribute to optimize the benefit of hydrangea, because the farmers in the Batu, they didn't take much the benefit on the apples, because the size of apple is quite small, and also the price is not so expensive. So they still take benefit from that, but they also focus on how they can get benefit from another additional plant that planted on the same orchard. Then based on that, farmer also get the benefit from hydrangea directly by increasing the number of functional group family and also promising the conservation of biological agents. Okay, maybe that's all. Thank you very much. So sorry, this is the entomology one.
Thank you, Professor Agus. Very interesting presentation. Now, I have advice to the participants that be careful to eat apple, because not all apple are edible. Okay. Yeah. Okay. And next speaker. Yes, we have Dr. Suradet Butchon. We welcome you to join us in here. Dr. Suradet Butchon, are you still with us?
Yes. Wait a moment. I start to sharing my presentation. So.
Okay. Yes, please.
Hello. Can you hear my sound?
Yes.
Did you? Okay. So first of all, good morning, everyone. So for me, the last person that I will give the presentation, then thank you for invited for keynote speaker. So, and in the first section is very good section that can introduce about the, thank you for Professor Alexandre that introduce about the compound and natural products for me. And thank you for Professor Patricia and introduce about the IBM integrated pest management. So first of all, I would like to introduce myself. My name is Suladet Putashon. I come from Department of Entomology, Faculty of Agriculture at the Hang campus, Kasetsart University, Thailand. So today I would like to take this opportunity to talk to you about the natural product and their biological activity. So I would like to introduce you about the many kind of the agriculture planning or etc. We have to try to show that what we can apply to pest control. So everyone know that we have many methods and the farmer or agriculture practice should the chemical control firstly. But nowadays we have many problems when we use the pesticide or the chemical control. So we have many tactics that can apply to the field, such as cultural control, mechanism control, physical control, biological control, and microbial control, and genetic control, and regulatory control. And the base that we use to control integrated pest management or integrated control, we cannot use only one method or single method to control the pest that interact in our plant or some kind of pest that very important in the economy plants, for example. So that to give me to talk in topic today because of the chemical control problem that everyone know that why we need the natural product because of the chemical control that the farmer apply to the field, such as like insecticide, herbicide, or fungicide that are used in the agriculture. And as you know that many problems that come from when you use the chemical, such as effect on non-target organism and pesticide resistance, secondary pest outbreaks, and environment contamination. And the main problem of the lack of trade, this is import chemical. And nowadays we are looking for the biological control that can be save the world and can be economic friendly, can be eco-friendly for the environment. So, and now we also looking for natural add like biopesticides. I give you an example like we can get the natural product from the high plants and also in the medical plants or wild plant that we can take a look what kind of plant that can be active or can use food for instead of chemical. And also we're looking for microorganisms that can produce many products and new product or novel product that can use in the agriculture. So, and I would like to show you about that. In Thailand, we have also the problem that we are import many budget that we pay for the pesticide, that chemical that we import to the country in terms of the herbicide, fungicide, or insecticide. By the year, or you can see in the table, sorry that in Thai, but you can see in the number. By the year of the year by year, the number of the import, the chemical that to Thailand, that the number increase every year. So that's why we are concerned about the environment and the cost to produce the product of agricultural products. So that's why this to give me to talk today. So, and if we talk about the natural product, and still have the several questions that remind to be the answer. Yes, right? How many natural occur compounds are currently known and how diverse they are? And what the most sequentially that the occurring the chemical support that of functional group or secondary metabolites. And with the main point of natural product for marine or for terrestrial or for microbial that both promoting source to new biological activity compound, because we're looking for the active compound. We can discover any new compound, but any new compound is could not active all, right? So, and what the proportion of the biosphere that's still explored, unexplored in terms of organism or in terms of their metabolic content. So, as you see in the photo on the graph in the figure 8, as you can see that the number of the natural product can isolate from the plants, fungi, animalia, bacteria, that in the past is many of the compound that was isolated, but I interested on the fungi that it could have like a different environment and could isolate the new fungi, and the new fungi could be produce the new product that can be active in the for the drug recovery or for the new bioagent for the agriculture practice also. And the graph in the picture B, that as you can see the number of the percent of the compound that, and also the classification of the compound that we can isolate. So this is just for example. So, and now I will talk to you about my interesting research, and I also work on that the natural product that I isolate from plants and also from the entomopathogenic fungi, and also I work on the soil fungi. And most interesting for me is marine derived fungi. So the last time I would like to talk to the methodology that how it works to get the fungi and how it work can get the natural product from our area or for our country. As you know, as you see in the map, the Thailand is located in the tropical area, so that's why we have many diverse organisms, and also we connect with the Andaman Sea, and we also have like a globe of in Thailand that we have very completely the environment, and we can discover the new organism that can produce the new compound. So I would like to show you that how the step by step, how to get the natural product and how to work on this. So firstly, we have to collect and identify where we would like to get the sort of fungi. This term I will talk like you can work on plant or you can work on the fungi or any microbial that you have to identify first what type of the fungi or microbial that you are interested in, because you can be take a look for the data in the online that have someone work on it or not. If still have a few data that you can be decide to work on this, because it can be sure that this you can find a new thing and can prove to the another thing that can be new. So the next step that after you collect and identify of the source, for example, this one I will deeply on the marine derived fungi. So after you get the like the coral or sponge or mangrove plants, and then you isolate and identify what is the fungi that you can isolate from that organism. And next step that you have to know by isolation or purify, you can use the molecular technique or etc. And then after you get the pure fungi, or you know what is the fungi name, or you can discover newer fungi on the organism that you isolate. And then you go to the fungi culture. Many techniques in the fungi culture. You can culture in the liquid media, solid media, or you can treat with different environment for either two the fungi can produce the new natural product or new secondary metabolite that the fungi can produce. So, and then you can try to extraction and fractionation by use the knowledge from the chemistry, while you can use the chromatography to work on it. After you purify, after you get the extraction, and then you can isolate and purify metabolite that you got it. And then after you know what is what is compound, and then you can go to the bioactivity compound evaluation. So the one I show you how we collect the sample. We go in the deep sea because in the deep sea have many strength for the salty taste and very deep, very pressure. So we are iterating that the organism that stay in very deep and very pressure environments can produce some novel fungi, and that fungi could produce a new compound or new natural product that can be active in the several area for the medical area or for the agricultural area. So after we collect, we identify organism, and we identify the fungi that we isolate from the organism, and we go to the step of the fungi culture. After we get the pure compound, pure isolate of the fungi. So in this photo is like the penicillium. So we culture in the PDA, potato egg for 7 days, and then we transfer to the solid media. In this case, we use the coupé light. And then we culture inoculate for 30 days. And we are isolate or soak in the ethyl state. It's an organic solvent for 7 days. And then we filtrate. And then we use the evaporator to reduce the pressure and get the mucus or the crude extract that we call the crude extract in the brown one, in the 11-year flask. And later on, after we get the extract, we can apply in the column chromatography. In the metrology is a chemistry area, so many techniques that and very hard work to get the compound and to know what kind of compound that we can isolate from the fungi that produce the fruit extract, right? So the first step that after we got the extraction, we apply to column chromatography. In my case, we use like an open column chromatography, and then we do like or someone can use the flat column chromatography. We do like a functionation of the column chromatography. Then after that, you have like what if fraction is contain many compounds. So we go to step of the isolation and purification of the compound. So firstly, after you have the fraction from the column chromatography, by each fraction will contain 1 compound, 2 compound, or very complex compound in the fraction. So later on, we have many techniques. We have to observe all fraction that we collect is like a solid form or liquid form or mixture form. Later on, we decide the method how to isolate and purification. In this case, if your compound can like a solid form, we can try to precipitate compound or recrystalline compound. Or if your function have like a 2 or 3 compound, we can use like a preparative thin layer chromatography technique to isolate compound. After you use several techniques to get the pure compound, then you send to the next method to know what kind of compound or to know what is structure of the metabolite that you can isolate. So we cannot use only one technique. We have to use several techniques to join together. For example, in this case, we use NMR. We use one dimension, 2 dimension to know metabolite that contain how many carbon, how many oxygen, how many nitrogen that contain. And if your compound suitable to be the feedstock, we can use the X-ray crystallography, or some compound that you know that we have like stereogenic carbon point, so we can use the ECD2 analysis, or some compound as you know from the information from NMR or information from HIMS that know that your compound is a compound of the composed of the amino acid. We can use chiral HPLC analysis also. And we also use the IR spectrophotometry to helping to structure elucidation. After this method, so we know what kind of compound, and so we know what kind of classical group of the chemical that we isolate. And then we go to the biological activity evaluation. So we can do many areas. For example, in the agriculture, we can take like a insecticidal activity, or we try in the acaricidal activity, or in the area of medical area, we can take for anti-cancer, antibacterial, etc. So is this a general method that you can find out in the general publication, and you have to try to do this. And some method it doesn't work for everyone, but you have to adapt it and take the basically of them knowledge, and you can apply in your work. And I hope that every step will be okay. So I would like to talk to you in my experience that I would take postdoctoral in Portugal for 5 years. So we isolate many compounds, and on that time I went to the natural product and medical chemistry group. So in this group, the objective of this research, they are searching for secondary metabolite that can produce from fungi associate different type of marine microorganisms from different habitat. And also we do the biological activity evaluation for anti-cancer, antibacterial, and antifungal activity. So in the presentation, I would like to show you about like 6 or 7 case of the natural product that we isolation. So the first thing that I would like to talk to you is about the fungi that associate from strong associate fungi. The name is Aspergillus candidus. So this compound that we can discover the 2 new derivative, the namely bis benzenoid and hydroxypyridine. And in total, that the fungi that we could isolate about 17 compounds, and we found 2 new compounds. And we are good luck that have 4 or 5 compounds that can active in the antibacterial. And you see in the presentation, the number of 7, 10, and 17 and 16 that can be active in the antibacterial. And the number of the compound number 9, 10, 11, 12, 16, and 17 and active in the anti-cancer. So, and this one we published on the 2018. And I also being this compound because as you know that I am an entomologist, so I very interested in those of compound that can be active in the agriculture areas too. So now we are working on this compound in the mechanism of for the insect. And the next fungi that this for the work of the master degree that I would participate in her work. So she also worked on the strong associate fungi Calaromyces, and she can isolate that 2 new compounds about the new ergosolol derivative and bis anthraquinone. And she also would like that the anthraquinone that can be active in the anti-obesity. So this work that we are looking for to prove to another step, like we can use this fungi can do the produce the anthraquinone that can be the people that who overweight and want to decrease the weight. So can use the supplement that can be joined with this structure of the new anthraquinone that can be anti-obesity. And the next master that I participate in his work, that he worked on the spawn associate fungi, Neosatoya, and like 2 species that he worked on, Phenylene and Sunode. So he could isolate that about 11 compounds. And then he found out the one new compound, we give them the Penicillin E, that show the activity is an active compound that show the with antibacterial. So it's a excellent work, another work. So and another master that he worked on the PhD candidate that I also participate in his work. So he worked on the strong associate fungi Penicillium irrubescens that could isolate many kind of compound, and he also found out the hormone derivative, new one, and then the the compound number 8 and number 9 show the antibacterial activity. And another PhD candidate that she worked on the SPONG also, the SPONG associated fungi Neosatoya called the Sinta. So she find out the many new compounds in the new polyketide and new benzoic acid, but she bad luck that any new compound could not become active compound. So in the anti-cancer or any antibacterial, but we still interested on new compound that could be active in the another area. And the last person that I participate on her PhD candidate that she worked on 3 associated fungi that isolate from microalgae, from sponges, and from mangrove associated fungi. And she could isolate many compounds. As you see that the white structure is a known compound and the red structure is a new compound. But she quite not good luck too much that she just found that the active compound is only one compound, the ECC, that the compound isolate from the Eurotium salivale that can be shown in the active, in the antibacterial. In terms of for the chemistry and for the medical chemistry, so we are move on to the natural product for the agriculture and biopesticide that I also took the compound that evaluate for the pest in the agriculture. So I took the compound from the Hypochera lactobacillus that we isolate. It's not a new compound, but the known compound like ergosterol derivative, and we evaluate for the Moch, the Pusella Hydrocella, that as you know that they are very easily to resistant to the pesticide. So we try to many activity and we could have the good result too. And another fungi that from the soil fungi, the Osatoya simensis, we also isolate many compound and they are like the alkaloid group that we know that the alkaloid group of any alkaloid group can be active in the insect. So we can try in on the mice also. So we take like alkaloicidal activity in terms of the contact toxicity or residual toxicity. And also we take for repellents also or inhibition and caching. We try to use this compound to know that can be active or not. And recently that we also use the derivative of ergosterol to compound 1 and compound 2, and also we isolate the hydroxypropane from the insect fungi that could be active, show the activity on the mice. In this table, we try to use the compound to control the spider mice. The name is Tectaticus truncatus, and the fourth compound that show that they can be like can be toxic for the contact toxicity for the mice, and also show the repellency for very high repellency for the dihydroxypropane that show 70% of repellency. So, and I very interesting for the natural product can use for the agricultural. And now for my project that I working on now, so we are looking for the one economic horticulture plant that is a coconut plant. As you know that in Thailand is the one country that can import the coconut for every country. So we have like the main project that action of the production capacity, product quality, and value added of young coconut, aromatic coconut youth basic research. So I work on the sub project that managing coconut mine to increase high quality productivity and reduce the crop loss. So as you see in this photo, before the harvesting the coconut, so we hope that we can collect the many coconut fruit during that time, but finally we find out that the coconut don't have quality. So the main problem is come from the insect and mice also. So in this project, we try to discover what is the case of the dropping of the coconut fruit during they are grow up before harvesting. So we find out that have one pest that is collagen pest that come to Thailand. So it can be case for the coconut dropping about say 60% that can make the young coconut dropping. So we use the many techniques because the problem of this pest is small. We cannot see by naked eye. We have to take it in the laboratory and see under microscope. And even that, we don't sure that this pest come from where, because we have like a local pest, but it's not damaged and don't have effect too much for the product. But this case is very effect. So we try to know what kind of mite or what kind of insect that come from where. So we use the two techniques to use in this research. We use like geometric morphometric analysis, and then we also use the molecular identification for see that the DNA of the mice close to which country. Conclude that the genetic of these mice close to the genetic that come from India. And for sure that we have many farmers that import the coconut from the India to Thailand. So that's why we have this problem. And this case we still try to find the solution to solve this pest, because this pest is plant quarantine list. So that's why when some kind of pest that say in the pest quarantine list is difficult to manage, is like concern about the many kind of policy that we have to import and export the product from Thailand to another country, or export or import the product. And in this project, after we know what kind of the pest that attack to our plant, so we try to use the biological control to control, because we don't use the chemical first. So in this project, we're looking for the entomopathogenic fungi to control this eleophagic mice. So we know that Persutella thompsoni can use several to control the mice, and many countries use this fungi. But as you know that for you control the pest, you have to know the environments, different environment, and the method can be used different. So we try to use pen isolate to control the eleophagic mice, and now we are on process publication, and we can find out this some of them isolates can be active in these mice. So in the last section of my presentation, that I would like to give you my experience about that. I not work only the natural product. I also start to work on the mice also that, and we have one project that study of mice Acara in Thailand. And as you know that in agriculture or in the entomology field, we know that have what kind of pest that very small and cannot see by naked eye. And the mainly four type of mice that contained in the agricultural field is spider mice, false spider mice or eleophagic mice, and bronze mice that can damage your plants. So, and recently I just worked this just like 3 years ago. So we can discover many new species that from the plants. So these are first publication that we can isolate the new genus and new species of the eleophagic mice from the plant that collected from in Thailand. And that you see that in the map that we collect like 5 location, and we give the name like sapphire, siamensis. and you know that we can see the relationship about the host plant relationship with the this mine. We use the geometric analysis to see how how they how is different in the species. So and we could not find out that in the same species could feed the same plant but different location. It could be different pattern. So we cannot find that kind of thing. So and the second publication that we also find one new genus and two new species from the medical plant, because I work on the medical plant too. So we have many pests that attack, and one of them that very important because we cannot see by naked eye. We can see only the symptom of the plant. So when we saw the symptom of the plant, it means that already damaged. So we find out that one new and two new species of the yellow mite from the medical plant too. So this is all of my presentation, and thank you very much for your attention, and thank you for the Ministry of Education, Culture and Research and Technology Universitas Brawijaya. Thank you very much.
Thank you, Dr. Bertrand. Okay, for participants, now we reach at Q&A session. So please raise your hand if you have question and you can deliver it by yourself. It seems we have some question virtually, but first, Bapak Ibu in here, you can deliver it first. So please raise your hand. Any question? Okay, please, Karin.
Terima kasih.
Okay, so I see the question on the chat box. So this is the first question that
Excuse me, I have another question here. Maybe we can first to Mrs.
okay.
To Mrs. Karinda to deliver it first.
Okay. So Professor Hagas can be first.
Okay, Pak Agus. I have a more question. You found Trichogrammatidea. So I'm wondering what species is the host of this Trichogrammatidea, and what a pity that you didn't find the parasite, the Actulcera.
Yeah, it's difficult questions from Bu Karinda. Yeah, actually now we are still conducting to identify in detail for each species. Yeah, but we group it already. Several species belong to the order and etc. etc. We will inform you in detail after all of identification already finished. Related to the, as you mentioned about parasite of, yeah, we will inform you after information will after identification completed. Yeah, now this research still conducting. One manuscript now submitted already, and hopefully will be published, but still focus on the family levels, get identify in detail to the species. After that, maybe we will confirm in species, then we'll inform in detail. But as specimen, we collected and we preserve in our laboratory, so maybe we can check in detail for each species. Another things. Yeah, we have actually we use a the trap. We just use a trap to collect all samples, but we didn't do such kind of direct observation for that, and also we didn't collect pest and then we rear and then try to collect parasite, etc. So based on that kind of traps, it's difficult for us to collect to get the parasitoid that come out from the its pest, etc. But from the yellow sticky trap we found, and based on the morphological character we identify, and finally several of them belong to the parasitoid in a group of Trichogrammatida, etc. But is still running, conducting for that. But so we got difficulties because we didn't rear the pest, and then we cannot collect that kind of things. An example of parasitoid, yeah. Mm. Mhm. Malay stripe is better. And, yeah. Yes, to get the clear morphological character after, yes. Yes, yes, yes. Malay stripe, pen trap, yes. Yeah. Mhm. Yeah, yeah. Yeah. Yeah, thank you so much. Good suggestions, yeah. Actually, we have 5 students now conducting the same research on that one now, but several students already passed, and still we are conducting to continue to identify, etc., etc. In case of Agratum, actually we didn't manage the apple orchards. We just identify that the farmers, all farmers in Batu, mostly they have 2 type of condition on apple orchard. The first one is intercrop by hydrangea, and another one apple orchard with the grassy ground cover. However, they didn't plant that kind of grass cover, just come up by themselves, yeah. So that is the reason why Agratum become very dominant in the condition of grassy ground cover there. So I will check, because as you mentioned that Agratum also attract several kind of parasitoid. We'll check, maybe we will try to use another trap to collect that kind of parasitoid, yeah, and pan trap maybe will be better, yeah. But in case of us, We also have several plant trap that we set up in that area, but we didn't collect. We didn't get. We found maybe already, but we will check in detail. So we'll find. We'll try to find that. But for the next research, maybe we'll try to focus on why agratum can also stimulate several kind of parasitoid, etc. Yeah. Thank you so much, Bu Karinda. Thank you so much. And scale insect. Oh. Oh yeah, yeah. Yeah, scale. Yeah, in case of Apple, yeah, Apple have a big problem. Previously, they have a big problem with the scale insect. Yeah. Yeah. We have a problem because we just use only trap. Yeah, we didn't check scale insect, and then we just put it. It's better maybe for the next. Maybe we'll try to collect that one and also try to get information detail about parasite that attack on scale insect. Yeah. Mm. Scale insect. Yeah, yeah. Yes, yes. Better. Better. We'll do that. Yeah.
Thank you for the question. Next question, committee, can you help me to open the question, please?
For me.
Oh,
Okay,
yes,
there are three
please.
questions. There are three questions. The first one related to the grass ground cover. Is the grass a weed or cover crop? Is close to be a weed. What I can do academically? Yes, actually, as I mentioned before, actually we didn't manage that kind of habitat. We just identify there are two type of orchard. The first one, orchard that covered by grass cover crop, grass ground cover, is actually that one mostly is wheat, is not cover crop actually, because mostly dominantly occupied by agratum. Another one is hydrangea. In case of hydrangea, actually is planted by the farmers because farmers also want to get additional benefit from that kind of hydrangea. And the second one related to the intercropping is hydrangea, blah, blah, blah. Term of volatile organic compound, flower color, microclimate, and also others. Yeah, it's related to hydrangea. Yeah, actually hydrangea can have many role to attract not only natural enemy but also pollinators. Volatile organic compound we didn't identify, but it also may be related to the volatile organic compound that can attract the insect. Another things, yes, the flower color also can attract the insect, especially for pollinator and also natural enemies. Microclimate, of course, because hydrangea also can stimulate the microclimate around the hydrangea itself and also apple orchard. And for others, yeah, that is the reason why we identify hydrangea intercropping related to the habitat management, because that one also can stimulate many things related to the habitat for the natural enemy, habitat for detritivore, habitat for the pollinator, etc., etc. Okay, for the three, color of hydrangea flower depend on soil pH. Yeah, this one also my impression, because when we check from the farmers, the flower color of the flower actually is not the same. It close to be related to the soil pH. When I check from the internet also, hydrangea have the range of the color. Sometimes they have the red one and sometimes they have the blue one. If they have the red one, it close to be the soil pH with value is less than 7. And in case of the pH higher than 7, mostly they also provide the blue one. In case of between red and blue, they also provide different color related to the purple or violet. Yeah, this is also information that I also collected from the publication and also website, that the color of hydrangea also related to the soil pH. And of course, the soil, the color of the flower also can attract insect to visit the flower. Yeah. So as we know that actually in case of pollinator, they also attract, they will be attracted by the flower because of two things. The first one is because of the color. The second one because of the flower is open or not. Mostly if the flower is open, it's easily for the pollinator to go inside and then try to get the food for them, etc. So at least two things. So, but I think there are many variables that can also attract the pollinator and also insect to visit the flower. Yeah. Okay, that's all I think.
Thank you, Professor Agus. Next question. Do you still have another question? Okay. I think this is for Dr. Sarojat Bhattacharya. Please. This is a question in recent years. Many development of botanical extract for pest control include use of plant essential oil and the delivery. Also developed such as nanobiopesticide. From your perspective and opinion, how the commercialization and mass production of this product? And number two, what advantages and disadvantages between bacterial and fungi secondary metabolite for biocontrol? And the third question is, how for development for mass production of these fungi and their secondary metabolite in next step scale, like bioreactor culture and culture medium environment, also extraction method? Okay, Dr. Sarojat, please.
Yeah. So can you hear well? Yeah. Can you hear on? Sorry.
Yes, I hear you.
Okay. So for the first question, as you know that how it become like a commercialization or mass production for this product. As you know that for many people, many study that working on like a plant extraction or plant essential oil. So, but it could work. It's work, but it's not too much that the many plant that can become like a commercialization. As you see in the commercial market, that we don't have mass product for this. Why? Because of the many specific of the extract or many specific of essential oil that could be controlled or could be controlled like a single pace is not universal like a pesticide. So that's why when we could have many products for this, but the people or the farmer could not doesn't take or doesn't shoot for the first for use it. And nowadays, people are concerned about the healthy, concerned about the cancer, concerned about how to plant for like organic plants. So now we back to use this kind of thing, but nowadays we could not have much product that become like a commercialization. And for the mass production, as you know that when the compound or some kind of extract that could extract from like one plant or one species, and like the next year you would like to extract again, it could be get the same content chemical compound inside because of what, like a climate change, some kind of temperature change, some kind of nutrition on the soil change. So can that plant or that plant that you would like to extract or you get the essential oil change also. That's why this kind of problem is could not how to say, like essential oil or botanical extract cannot align to the commercial because it's not stable, for example. I don't know if I could answer the that what you want in the first question. Or for the second question, if you ask for the advantage and disadvantage of the bacteria or fungi, that for biological control, as you know that for the bacteria or fungi that they have the specific for use in the biological control. For example, this bacteria or this fungi can control only the bitter. So you could not apply in the okay, can apply for the moth or butterfly, but the most specific is a better way that you can use for the bitter paste, for example, that can see easily, but actually you can try to use for control any insect, but mostly bacteria or fungi, they have specific with the host. This is a disadvantage that why for the metabolite or from that fungi or bacteria that have this problem, and also they cannot tolerate with the environment. And now the climate change, the high temperature, and we can apply in the early morning or in the evening, but farmer it it doesn't like it have many protocol how to use it. That's why it's many disadvantage. So that's why, and for the advantage of bacteria or fungi secondary metabolite, for the advantage is that they have specific. That's why is can kill and can make the pest cannot be resistant because of them specific. So that's why that pest is cannot resistant of the metabolite of bacteria or fungi. And another advantage is you can find out in your country and you apply in your country, because mostly the bacteria or fungi that can isolate in the same environment and can develop and can use in the same environment is better, can be active. But if you import from the another country and that fungi or bacteria have different environment, and it could be active also, but who know, we don't know, but for sure if they are the same environment they can be active, for example. And the last question, how to develop mass production for the fungi and their secondary metabolites. If I don't know in the participation that who have experience about the extraction of the fungi or bacteria, that as you know that the compound or the extract that you can produce is not smart, that you have enough to test in the nursery test or the field test, but you allow you to test only laboratory test. So that's why when we already know what kind of compound that active, so you have to pass to another step like a synthesis, chemistry synthesis, because you have already model of the structure, what kind of structure that can be active compound, and then you go to the synthesis. That's why the secondary metabolite is some kind of the chemical, but they are, we can call like a green chemical or biochemical that can be new structure or can use instead of the some kind of the pesticide that used long time ago, and the pest already listed that. But if you concern about the how to make the production, and you know that we cannot use in terms of the secondary metabolite, we can use in terms of the fresh culture. Nowadays have many products in Thailand too. So if we know this kind of bacteria or this kind of fungi that can be produce the active compound and can be kill the insect, so we use the fresh mass production. We can mass a big amount of the production depend on the equipment. For example, I have a small autoclave, so I can produce 100 bag of the fungi that go up in the solid media, and that in that phrase culture we can apply in the agriculture. We don't use much for the secondary metabolite to apply in the field because we don't have much. We only know that this fungi can produce the toxic or something that can kill the insect. So let the mechanism work in the environment. That is the way that we can solve this problem, that we cannot produce a ton of the secondary metabolites. So these are all the answer. So I'm not sure that is the answer that you want to read. So I am not sure if Professor Alex can explain about the secondary metabolite that the microorganism produce. Is as to know that they can produce different anytime. Even you culture the same fungi, the same environment, but they can produce different compound.
Thank you.
Thank you.
Can I add additional
yes, of course.
information
Yes.
to Bu Karinda? Yeah. I get from my yes, I got from my the raw data. We have about 1, 2, 3, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It's about more than 12 parasitoid belong to the 12 family. Yes, as you mentioned that braconid also involved. Braconid mostly found in grass, braconid, compared to the hydrangea. In case of Trichogrammatidae, we didn't find from the grass ground cover, but we found mostly in the hydrangea. We still have several, as you mentioned before, about Encertidae, Eucolidae, Eleutheroidae. We have several group of family parasitoid. Yeah, but it's still in the raw data here. So thank you.
Okay, thank you, Professor Hangus. Various information that we can get from this topic, but I think enough for this session. So thank you for the speakers and also the participants for the amazing discussion in this session. For the next, so please, Master of Ceremony, to continue the session. Thank you.
Okay, thank you so much, Prof. Agus and Assistant Professor Dr. Suradet for the insightful presentations. And thank you for the discussion, Ms. Pralita. But before we conclude this event, I would like to invite the Secretary of Department of the Plant, Pest, and Disease to give a closing remarks for these events. For Dr. Iris Satrianti, please come forward.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good afternoon, ladies and gentlemen. As we come to the end of this seminar on innovative technology in integrated plant, pest, and disease management, I would like to express my deepest gratitude to all of participants for your participation and contribution. And also I would like to send our special thanks to all of keynote speaker, Professor Alexandre Soares Rosado, Professor Patricia King Jiha, Professor Agus Tarno, and also Assistant Professor Sudaret Hutajon. Your insight and expertise have been invaluable in this seminar. We had privilege to exploring innovative technology and integrated approach to manage the plant and pest and disease. I hope this knowledge and this connection we gain today will lead to fruitful collaboration in the future. So I hope today's seminar will be the start of many more collaboration between Fakultas Pertanian Universitas Brawijaya and King Abdullah University Science and Technology, University Putra Malaysia, and also Kasetsat University Thailand. Let us continue to work together to push the boundary of science and improve integrated plant-pest disease management. By the end of this event, I officially close the Seminar Internasional on Innovative Technology in Integrated Plant-Pest Disease Management. Thank you and hope to see you again in the next international seminar. Good afternoon.
Thank you so much for the Secretary, Ibu Dr. Iris. And thank you so much for all of the speakers for the valuables and insightful presentation, the materials, and I hope it will be a valuable insight too for the participants. And thank you for all the moderators and for the committees for the hard work to make this event come true actually. But before we conclude, we have a few announcements. Now we would like to present a souvenir as a token of appreciations to our distinguished keynote speaker. We kindly ask Prof. Alexandre Soares Rosado to come forward and Dr. Lukman Kuloto Aini to come forward to deliver the souvenir. Please come, you may stand here. This is the certifications to Prof. Alexandre. Thank you so much for being here with us today. Okay, 1, 2, 3. Okay, thank you. Thank you so much, Alex. And the next, I would like to invite Professor Dr. Patricia King to receive souvenirs of appreciation. You may stand out here. And Ibu Dr. Irisa, as the Secretary of Departments, you may stand up here to give or to deliver the souvenir for our keynote speaker. Okay, 1, 2, 3. Okay, smiles. Okay, thank you. And this is the certificates. You, Dr. Patricia. Okay. And for the next, I would like to invite Professor Dr. Agustarno to come forward also to receive souvenirs and the certificates. Wait a minute. No. 1, 2, 3, smile. Okay, thank you so much. And for Assistant Professor Dr. Suradet Butakon, we have a certificate of appreciation for you. And here are the certificate, and we are really appreciate your times and your contributions for our seminar valuables. Okay, thank you so much, Dr. Butakon. Thank you so much. Have a good day in Thailand, Dr. Suradet. Okay, so thank you so much, all esteemed speakers, ladies and gentlemen. I think we are almost at the end of our events today. And thank you so much for all of the participants, and thank you for being here. And I think that's all for today's. And I would like to tell some informations that we have arranged a luncheon for the guests and for the lecturers of the Plant Pest and Disease Departments, lecturers and professors, and all of the invited guests. Please have a nice luncheons in front of the Senate Hall. And for all of the participants, I appreciate your times for coming and join with us today in this international seminar. Thank you so much. And I will end this event, and have a wonderful afternoon, and see you at the next scientific event. Goodbye, everyone. Wassalamualaikum warahmatullahi wabarakatuh. Bye-bye.